Okay, so let's get started. Thank you very much for all of you being here. Also, thank you for the speakers, for the organizing committee. You will be seeing them throughout the day. They will be introducing different speakers. Um, so this is a joint effort between the UC Davis Coastal Marine Sciences Institute, in which I'm part of, Muse is part of, and others in the room, and the Delta Science program. I see Edmond around there. I see different people. You will be like, we're several, so I prefer to just keep it short. For this symposium, 1 day symposium, this has been done for over many years. I think that the first one was 2017. Once a year, we have this symposium. Today is going to be about food webs in the Delta. And we have a very good lineup of speakers, and we have organized this symposium in 3 sessions, and we're going to start with a couple of keynotes that are going to actually do the keynote together, like one first, then the second. So it's a treat to have them here. I will then introduce them later. And the 3 sessions are about modeling. So we have 3 speakers that are going to talk about models that address ways of using data to capture complexity, uncovering hidden relationships, and generating predictions across large scales. Then we will have a break. We will have a session on empirical studies that will feature recent site-specific studies that provide generalizable insights and inform modeling. And then we will make everything actionable in a 3rd session that is going to be about management strategies, which will feature applied research that offers novel solutions to food web management. Before the 3rd session, we will have lunch. It's not provided. We will have a work lunch among speakers. We're gonna give them food, but they're gonna work together because we are gonna have a panel after the session 3. That's the last part of our symposium. That panel will be structured with questions you will provide throughout the day. So look at your tables. On your tables, you have pages with the program and also with a barcode. We are using technology on our favor. And the idea is that throughout the day, you can, of course, ask questions to the speakers in the time allotted. But if you have like deep thinking questions that you don't feel like asking them in 10 minutes that we have after each presentation or less, just write it there. You can also vote for the questions you like the most. We are organizing them. So Sophia, right there, Sophia Simon, my student is, PhD student, is collating the questions and on working and taking notes and all that stuff. So we will be working on a very structured panel at the end to really have deep conversation, deep thinking, critical thinking, and also engaging with you guys, the audience. Then also I want to emphasize that we have we know you register and you provided your information during registration. We know we have a lot of expertise in the audience as well about the system. So please feel compelled to also chime in your answers. So if you feel that you're an expert of the question, yes, please also answer, raise your hand and answer, or submit it as a question, even if it's not a question. So we really want this symposium to be more a working group, like we are all engaged in discussion. Okay, so let me see. Looking, I'm not missing anything. Yes, I think I covered everything I should have said. And now I can introduce the keynote speakers. I want to emphasize that the idea is that the keynote speakers are the experts of the system that are going to give us this introduction to the system. And then the sessions are going to be the ones focusing on these different aspects. So our first keynote speaker is Liz Marie Windham Meyer. She was previously a university professor. She's a system ecologist with the U.S. Geological Survey, but now she's a lead scientist in the Delta Science Council. She has a PhD in ecology, evolution, and natural resources from Rutgers University. Her research examines processes at the interface of land and aquatic systems, including water flow, storage, and quality at landscape scales. She has extensive experience in the conditions in wetlands and estuaries that sequester and release greenhouse gases and control methyl mercury dynamics. Also, our keynote speaker is Matt Young over there. So both are Liz Marie and Matt Young right there. He's a research fish biologist with the U.S. Geological Survey. And he works in a variety of systems ranging from the Pacific Ocean to the high desert, exploring relationships between native fishes and changes to their environment. This includes the spread of non-native species, habitat alteration, and the loss of underlying ecosystem processes and the potential for habitat restoration to provide key benefits to native fishes. So please, Liz Marie, and I don't know if you want one after the other, like, or...
All right, this is my first FoodWebs workshop. This is fantastic. I'm looking forward to it. I really believe in cross-disciplinary work. I'm grateful to the organizers for inviting me to present this morning, and I'm also especially grateful to my keynote presenter, Matt Young, for helping me work through a plan that could help pull together, again, this concept of physics to food webs. And of course, we're using this USGS product as a framing because it so well pulls together synthetically data on that's crossing scales, that's crossing disciplines, and then also has management implications. So we're hoping that in today's talk, we get across three main points. Our three main points really are the importance of syntheses. It's important for food webs. It's important for carbon cycling, which is where my focus has been historically. And it's also important syntheses cross systems. So we all think about the San Francisco estuary being unique, but we have a lot to learn by looking at other systems. That's the first. The second is models, and how important it is from the conceptual to the numerical to the forecasting that we understand our biases when we have models, and the fact that they're limited by observations. So again, the importance of observations. And then finally, the idea of getting us out of our boxes. It's very easy to get in, and I think we learn a lot when we step out of our boxes. So those should be some themes that you'll hear in my talk as well as Matt's. So I think the first point I want to make is that if you care about fish, if you care about carbon, if you care about humans, then you care about water, and it's across the whole landscape. This is a very interconnected system. And this water cycle that was revised in 2020 by USGS, the key points about it are that humans affect nearly every aspect of the water system. So it's not the water cycle of your grade school days. We're really appreciating the role of humans now affecting fluxes, affecting how much groundwater recharge there is, how much evapotranspiration there is, how much of that water actually still makes it out to the estuary. And another thing I wanted to highlight in this graphic is the nice little delta here, which is let's see, I don't know how to make the light come on, but that's okay. I think you can see the little delta. It's right below urban runoff, right there. That's a forward-looking delta. That's the ones we all tend to think about. And yet we have really much more of a backwards facing delta. And it's because of this choke point, of course, at the Carquinez Straits. So the reason it's important is because our formation is a little bit different and we have different issues than other deltas. But on the other hand, we have the same general problem of subsidence and some water quality issues within the Bay Delta framework. The other thing to point out here is that water quality is dynamic in our system. It's not a single fixed issue. It changes through time. The most obvious one to think about is salinity intrusion and how we manage that saltwater wedge with different techniques to keep the delta in a fresh frame. But the point I want to make about water quality is that it's not just a function of transport, the rate at which water flows, which I think we tend to focus very much on transport for water quality. It's also the rate of reactions in the water. And so, for example, this very simplifying mechanistic diagram that shows the importance of timescales was shared with me by Lisa Lucas about 10 years ago. And it really stuck with me about the importance of understanding the rates of both of these, because that's going to really control what levers you have to improve the system. We actually developed this for this originally for methyl mercury, and that was with a project with Mark Marvin Di Pasquale at USGS. And what we realized was that a lot of the methyl mercury transport in the Bay Delta was actually driven by transport, not so much all these mechanisms that we all get into the details of how fast those, or how important all these fluxes are. Transport explained a lot of the distributions of methyl mercury in the system. And I think that's the key point here is that you do not forget about transport, even if you're a biogeochemist. Okay, a very, I would call it unuseful simplifying mechanism is our boxes. And I think we like to think about working in particular parts of the Bay Delta system instead of the system as a whole. The primary issue the reason we put ourselves in boxes is to reduce our variability, because we understand that system. We understand its constraints, and we have a literature to go to. But it also limits our solution space to only think inside that box. So getting away from regionalization is one advance that I see happening, not just in water quality measurements, but also in things like the harmful algal bloom work. And then I'm seeing it happen also within the food webs community, which is really makes me happy to see. So getting back to timescales, I think an important one for us to always step back to is that there's a lot of things that have happened here over the past 18,000 millennia when we started having sea level rise. It used to be, the Farallones used to be 19 miles off the coast. You could just walk out there. And when you look at sea level rise, of course, it was very, very rapid until about 7,000 years ago when it started to flatten out. And so the two rivers all had come through the Carquinez Straits, that choke point, but then now we've been backfilling that whole system with not just sediment, but also peat within that basin that's at the inner side of the upper San Francisco estuary. Consequently, that time frame of about 6,000, 7,000 years ago is about the same time that cities started showing up on the global map. So this number, 6,000, 7,000 years ago, is fundamentally important at a global scale in terms of developments around the world, of coastal cities in particular. Another point to make about the Delta space is that we, again, frame it as this distinct unit, but the previous occupiers of this land, the previous owners of the land that we're on, they actually thought in a much greater way of this connectedness of water across the landscape. So not really focused on specific locations, but on how the water was moving through and being the populations that they fished for and so on were obviously extending far beyond where their home territories were. And this gets to the issue of boundaries. Again, the more that we set these boxes and these boundaries, we have less solution space for the challenges that we're facing in terms of water quality as well as food webs and key biota. So thinking about the Delta today and what we've got, what do we got here in our backyard here? It's obviously a very large bowl. I don't use the word hole anymore. I use bowl because that's really what it is, and it's basically hollowed out, and it's lost about 100 teragrams of carbon over all of this, over the past 150 years, which is just it's a huge amount. It's enough to have an atmospheric signal on an annual basis. So it's actually a really important sink that you can see the numbers play out. I'm sorry, through carbon oxidation of the soil. So it's an important loss term as that system has hollowed out. And then when you look at that same graphic on a landscape, you can see that it was the farming that brought us this big bowl is still there. It's still ongoing. And there's some landscape decisions that have to do with what to do with this farmland. But it's also it's so subsided in some places that it constrains our future options. So land use decisions that we're making are ultimately about reflooding and where to put water. That's ultimately what these decisions being made right now are. And we have a lot of constraints because of past land use. So let's just talk about the past briefly. The very nice historical ecology work by Alison Whipple and others at San Francisco Estuary Institute clearly showed through one way of knowing, through all these journals and reports and other documents, even stories from Native First Nations communities, they were able to put together what the Delta looked like pre-human, I'm sorry, that's not what I meant, pre-colonization in the 1860s. And so it's all fresh, basically. And we know it's also fresh because Judy Drexler amazingly collected a ton of 8-meter deep peat cores across the Delta, which just yielded a tremendous amount of information. And so one of the, I'm not sure if my little graphic, there it is. So I just circled for you from Browns Island up to Bacon, the Bacon channel there. And so what you can see is that basically, if you go back in time, 6,500 years, looking at the first graphic, it's always been fresh at the top. And then only recently, within the last about 2,000 years, have you started to see a signal of oligohaline, a little bit of salt showing up at the top of Browns Island. The last point at the bottom is the uncertainty in that, and that's just within the last 200 years. So the issue is we know the system has been historically fresh, perhaps some excursions, but they don't show up in the peat record. And so having the return of continuously salty water probably will change the system in a significant way. The Delta today, of course, no longer looks like that huge freshwater marsh. We now have dominated by agricultural land. And of course, all that productivity, this incredibly warm, wet system that made so much productivity for our food webs is now gone, and it's been appropriated for humans. So it's now crops, and it's still fresh. The system's still fresh, but it's an artificially managed fresh system at this point. But it's still productive, it's still fresh. And then I also want to highlight that it's diverse too, in a human component in the types of communities that we have. And I want to highlight that the Public Policy Institute of California's 2024 report that just came out on California's freshwater biodiversity highlights the Delta and some of the ways that we could protect diversity in the Delta, particularly for fresh ecosystems. So that said, one of the ways to protect is to do tidal wetland restoration. And so a lot of this work on tidal wetland restoration is focused on the idea of reflooding and getting this system back in where you can grow soils. And what's so special about tidal wetlands is they don't saturate with carbon. They just grow and grow and grow and grow as sea level grows. And the only problem is if you hit a tipping point where the system cannot keep up anymore with the sea level rise. The other point about this from a carbon perspective is the soil volumetrically is dominantly carbon. So carbon fluxes play a huge role in the sustainability of future landscapes in the Delta. The other thing to point on the right-hand side is this, there's a basic mechanism to think about, which is when we talk about sediments, they are important. They become increasingly important to the accretion rate as the sea level rise grows. And that's partially because it's getting more underwater. The system is getting having a higher load of mineral because it's poised lower in the frame, but also the rate to keep it up, organic matter can't do it at a certain point. So the place in the Bay Delta that has almost the most data of any tidal wetland is Rush Ranch for carbon fluxes. And that's because USGS established an eddy covariance tower out there in 2014. So it's 10 years of data on carbon dioxide and methane fluxes. And then it's coupled with the first Mallard station, which I know many of you are familiar with. You have different questions that you ask using the data from First Mallard Station. What I was so impressed with from the food web people was how much you've thought about all of the weird little channel configuration issues that we think about now for carbon, but you were ahead of the game. And I think we learn now from your work about how we can use this data to understand carbon fluxes. One of the key things is that if you look here on the bottom right, you have just a comparison of how leaky our system is. We produce a lot of carbon. Let's just look at the yellows, tidal marshes. There's high productivity, but we also have high loss. So the amount of, it's a conundrum you have to get around, which is basically the systems are so productive, they store a lot in the soil and they export a lot of carbon. That's how amazing these systems are. And that export is something that's only recently starting to be considered. So the difference again in a neap cycle and a spring cycle is really fundamental for you for productivity in the channels of phytoplankton, for example, but it's also fundamentally important for the fluxes of carbon on the landscape. So this very, very nice animation that Paul Sumner shared with me isn't playing. Don, well, thank you so much. I just want to point out that many of you have been out at this site during flood cycles, especially the really extreme king tide. I'm always on the land and you're probably in the water. But from the land perspective, what's not playing, what you can't hear is the soundtrack. It's like a cascade of little waterfalls. I mean, there's all of these channels getting flooded, particularly on the king tide, getting flooded, and then on drainage, you hear it coming out, dripping out. And it's all of this basically water that's laden with dissolved inorganic carbon, alkalinity, dissolved organic carbon to a lesser extent. And there's a particulate organic component, but it's actually very small compared to those dissolved. So the importance of this exchange is not just for fish, it's also for the fluxes that are coming off the landscape and also for stabilization of carbon on the landscape. Thanks, Ben. How do I get Ben? Thank you. So on towards stabilization of carbon, I think this is the kind of the taking it from the macro all the way down to the micro. One of the best things about looking across disciplines, you start to see people have figured things out elsewhere. In terrestrial world, it was discovered that soil carbon stabilization is actually, I'm way over, aren't I? Oh, thank you, Moose. Okay. A lot of the carbon stabilization, we talk about carbon stocks being large in the soil, is not because it's too cold or it's too acidic or it's lignin or something like that. It's typically microbial access to the carbon that's in the soil. And sometimes when you step back, you can start to see that what's old is new again. Darcy's Law, which we tend to focus on carbon being exported, how much carbon is coming out of the system through groundwater flow, for example. The stabilization of carbon in the soil has to do with flow as well. So what that means is that if you have high flows of, I'm assuming this is the button, so yeah. If you have high flows of water, you'll have basically high aerobic respiration rates, high turnover of the carbon that's in the soil. And then when you have low fluxes of carbon, so for example, deep in the soil, you won't have very high aerobic respiration. Instead, you're going to move into methane production, but you'll also have high amounts of carbon preservation. So ultimately, flow is controlling, Q is controlling carbon in soils as well. I think this is just important for us to step out of our boxes to see these kinds of things that we might not have seen because we're so focused on the details. And this is important, again, for radiative fluxes, because a lot of times people talk about reflooding lands for the purpose of climate mitigation. And there's a lot that we can do there, but there's what I'm going to propose is it's not just a matter of how much CO2 is going in, how much methane is going out. It's also how much is leaving the system. And this flux here is very strongly influenced by, for example, channel geomorphology, or I should say geomorphometry. And then also things like ocean temperature and so on. And this is being modeled right now through a couple different projects. And I will keep going. I'm happy to talk about radiative forcing another day. I think the other point here is just that brackish marshes are sort of amazing in the sense they have high carbon sequestration and low, virtually no methane production, whereas tidal freshwater marshes have high productivity, but there's a large methane component still to be concerned about. So I'm going to start to wrap up by saying that there's many challenges here, and they're coming in many different directions. The whole system is connected, and I think once you start to step away and see this, you might be more happy stepping out of your boxes. The ocean is changing, it's rising, it's hotter, the upwelling patterns are variable. We don't control precipitation per se, but we do control where it goes. The wildfires on the landscape, a thirstier atmosphere, things like that are causing changes in our freshwater flow in. And then from a groundwater perspective, we're altering and in some ways, some places enhancing groundwater management, but mostly we're not. Mostly we're draining the groundwater. And we have legacy contaminants and emerging contaminants. And there's lots of challenges coming into the system that I'm not trying to make it complicated, but I want to highlight that they're important to consider. And then finally, there's ways of knowing. And I want to highlight the need to just not have a hammer where everything looks like a nail. Use as much data as you can from a variety of perspectives, because across scales have richness of data and possible interpretations that you might not have thought. For carbon, this has become really important. A new, really game-changing thing on the table right now is this SWOT, so surface water ocean topography, SWOT. The new satellite that went up, and they're going to drop the data for public consumption in July. And this will be through a USGS website called WISP. And I think there'll be other places too, but that'll be a good one. And basically, this is the scale of 1 centimeter elevation over 1 kilometer. So millimeter scale measurements of water elevation from which you can calculate flow. So ultimately, you can get to discharge. It's pretty amazing. And this is how we're thinking about doing ungauged rivers, because now we have data sets that might be applicable there. And this image, Toby Minear sent from there. And the final thing I want to talk about observations is this is the oldest way of knowing. I mean, the only reason we know anything is because we make observations and we record them. The Delta Residence Survey that came out last year is one entirely new data set to think about with observations from people who live in the Delta. And I want to highlight the ability to use that. Another way of knowing is modeling. Lots of different models out there. Love to start working with them all together. This is work from Noah Knowles. He presented it at the Bay Delta Science Conference in 2021. I highly hope that you all are considering going to the Bay Delta Science Conference. It really is an opportunity to explore things in more detail. And he was able to, in this one, use the cascade model, which is, again, a highly interlinked model of models, where each model is owned by somebody who really understands it well at a first principles level, and then pull it all together to make these large statements, such as, this is an estimate that Fred Fire gave him a 24C boundary for smelt mortality, and to see how, under future conditions, if we have really sort of the worst case scenario, warmer, drier conditions, we'll be up to about, in the Central Delta, upwards of 100 days, or 2 months worth of days at which 24C is exceeded in the water column. So these kinds of projections are possible. And then the final thing I want to highlight is synthesis before we step into Matt's work. This data here, this was something that had to be done. There was no national map of Z star, basically relative elevation of tidal wetlands compared to sea level, and particularly the sea level range. So where are you in the tidal range? And so we created this in 2022, a map for the country. And one thing that became really clear was where are blue basically means low in the tidal frame, and red means high in the tidal frame. And everything that's colored here is a tidal wetland. But you can see for us in Suisun and San Pablo Bay, we have a lot of blue, so we have a lot of subsided lands. And they also tend to be back from the coast. We have peat soils that have oxidized, as opposed to, for example, Terrebonne or Louisiana, where the blue tends to be on the water's edge because it's right near the water. So it's just sort of eroding away right on the edge. It's a different kind of pattern. And then the final synthesis of concern to me is that we never get included in these Delta synthesis projects where they take 50 plus sites from around the world. So I put a big red circle, and I really would like to see the Delta included. It's because we have a backwards Delta. It doesn't work the same exactly to fit their system. But we do, if I look at this sort of Pasteur's quadrant of societal risk and physical risk, we are definitely up in, let's see if I can get this. We are definitely in the upper right corner here, when you start to look at the sea level raise, the relative sea level rise rates that we have here. It's not about 3 meters at the sea, it's about the subsiding land. And so interior subsiding land, so in some cases, there was a great talk yesterday by Stefan Talke. He also has a Nature paper recently that shows 13 centimeters of loss per year is what we're talking about in places like Rio Vista and some other places in the Delta regions. And that is the last thing. I have no graphic here to show you, but I do want to highlight that I highlighted food webs twice now at the lead scientist report for the Delta Stewardship Council. They loved it. They were very interested, and they were particularly interested in the synthesis, the way that you all, and the papers I highlighted were, I think one of them, Sam is actually talking today, and the other was Rosemary Hartman's Dry Me a River paper. They loved the idea of taking all that data and saying something with it over a long trajectory, and then point us where to go from here. So I just want to highlight the importance of synthesis, particularly for your work, but really across all of them. And then I think the final slide is just to reiterate, I think we need to work together. The challenges are too great, and none of these are beneficial to study alone. They have strong connections. This particular graphic here, I'm going to highlight again. This is what HABS monitoring looks like today, and this is what HABS monitoring is going to look like, hopefully, when the MURHAB project is complete in 5 years. They're trying to basically streamline approaches, everything from the remote sensing to SPATs for toxins, and try to come up with a system to understand how these work together, so that we can have a cross-system assessment. And I went way over my time. I'm so sorry, Matt. And I think we'll move to Matt now.
Hi. Can you hear me? I want to start just thank you for being here. Thank you for inviting us. This is really exciting. We worked together to pull together sort of an integrative story, and this is our inflection point. We're going to switch from this discussion about the delta, the system at large, and a lot of these physical processes at scale. And we'll talk about food webs for a second. We're going to go over time and it's going to cut into questions, but there's going to be a lot of time to do that throughout the day. So, sorry. And in the panel at the end. So if you have something really pressing, we would love to talk about it, but I'm just giving you a heads up that this is going to run a little long. So when we talk about food webs, when you hear the word food web, a lot of people think of this conceptual model, right? The old ball and stick model where community elements are represented by balls of various sizes and connected by sticks that have generally represent some sort of energy transfer, right? Relatively straightforward conceptually, but super complicated and not necessarily intuitive. Food webs themselves, it's like entirely a human construct. It's our own conceptual model for how we deal with how complex ecosystems are. There's a lot of ways to think about them. There's a lot of ways to talk about them. There's a lot of ways to study them. So I zoomed way back, and we're just going to look at a few of these like different ways to think about trophic ecology and the interconnectedness among communities. So we have a few straightforward questions I'm going to talk about. They're not simple, but they're very straightforward. And we can use food webs to think about, who eats what, where, right? Really straightforward, but really crucial for a lot of reasons. Where does carbon go? So Lisa Marie just talked a lot about, give an excellent primer on how carbon cycling can vary on various spatial scales and how it gets transported, et cetera. And we want to think about this in a food web context. And then how does the ecosystem work? This is like, obviously a straightforward question that is not simple, right? But we can get insights if we think of, from food webs and the way we think about it. I'm hyper caffeinated. So, if you need me to slow down, just tell me like, hey, chill, dude. So who eats what where, right? So we're interested ultimately in higher level consumers. This is one of the goals of the symposium is talking about food webs and fisheries management. Consumers eat things. They eat fish, or they eat bugs, or they eat plants. Some weird fish eat plants. One of the most infamous consumers in the San Francisco Bay Delta in its entirety is striped bass. Many of you are familiar with this. Striped bass were introduced into the system 150 years ago, and they have had a tremendous and largely unmeasured impact on our system as a whole, right? This lovely thought piece, which I highly recommend you read, by Matt Nobrega and Will Smith, just talks about how the introduction of this predator fundamentally affected delta smelt, and most of that impact happened before we were looking. Striped bass are a super interesting example of this top-down control on ecosystems, because not only have they had substantial impacts on native fish we care about, but they're insanely flexible. So striped bass eat different things at different times, depending on what's available and what the conditions are. And if we don't have an understanding of how striped bass diets change seasonally or across different habitats, then the way we think about them and their impacts on the system changes dramatically. This is one of those things where, modeling and synthesis need to be informed by relatively refined observations, okay? There are many, many examples of this concept. If you want to talk about foraging ecology in a different sphere, right? If you're interested in something other than striped bass, Abby is going to talk about salmon later, and salmon in the ocean are more fun than striped bass. Just throwing plugs out. There will be a lot of these plugs, so hope you don't mind your pictures, those of you who are speakers. So where does carbon go, right? So I'm going to just sort of give vignettes and ways that we can think about using food webs for management questions. Now we're talking about carbon, right? So primary producers photosynthesize. It's like the greatest evolutionary adaptation in the history of planet Earth. It's crazy cool. And we want to know how it feeds food webs. So we have primary producers. They get consumed. Carbon from these primary producers gets consumed in some fashion. And then that trickles up through the food web, right? And ultimately what we're interested, or one of the things we can be interested in, is how these different photosynthetic pathways or primary producer pathways are structured across the landscape and result in benefits to fishes. In a really simplistic sense, we can just look at a fish community, the total biomass of a fish community, and see where its carbon came from. And this varies on the landscape. Different places have different primary producers, and you can see that in their fish communities. For example, in Rush Ranch, with a very healthy intertidal wetland with a lot of emergent macrophytes, that's what the fish ultimately eat, right? Most of their carbon is the green. Is the green. In other places where there are different producers, these ratios change, right? So just thinking about how this changes at small spatial scales on the landscape, and then how transport dynamics and scaling out would result in different interpretations. For the latest on cutting edge tidal marsh ecology, Megan will be talking about that later. Wow, that background is terrible. My bad. So this is just sort of the spatial context on the landscape, the actual physical template within which food webs happen. But in rivers, estuaries, and the ocean, water moves. And so there's a dynamism here that is, really a complicating element to this. So, we looked at these sort of food web dynamics in the Sacramento Deepwater Ship Channel. For those of you who aren't familiar, it's in the Delta. It is the simplest possible physical system. It is a long canal dredged for shipping with very little structural elements to it. But it has very strong tidal energy gradients that result in really stark differences to food web structure, where pelagically derived carbon really matters in certain areas, and then benthically derived carbon that came from things like macrophytes or microbial loop processes or other things matters in some of the accumulation zones that occur due to tidal energy, right? So in addition to this spatial structure tied to the physical template of the landscape, tides can really affect how carbon pathways work and food webs are structured. And if you want to talk more about plankton, Brian and Kyle will be late. How does the ecosystem work, right? This is like the literally like billion-dollar question. And there are many ways to explore this. There's been a lot of work looking at this in terms of complexity of food webs, in terms of interconnectedness of food webs. And there's a lot of recent work looking at top-down controls in food webs. So if you have a critter, is its abundance dictated more by bottom-up processes? So how much productivity is there versus top-down, how much is it getting at, right? And coming back to our very simple laboratory, a colleague did an excellent analysis looking at the relative importance of top-down and bottom-up controls and how that can be structured by these tidal energy elements, right? So this tidal energy is a huge part of how this system functions, and is something to be taken into account. Oh yeah, I want to point out, remember this like rate of transport, rate of reaction thing? This is like a perfect example, right? In high exchange environments where transport is high, the reaction rate isn't fast enough to dominate. Versus when transport's low, reaction dominates. Anyway, just like you guys thought we weren't going to be self-referential. There's been a more holistic discussion of top-down and bottom-up controls in the estuary. That'll be discussed later today. Excellent work. Many of the co-authors for that are sitting in this room. There's also other ways to think about these ecosystem processes, where there, in addition to top-down and bottom-up, there's biotic and abiotic controls that will get addressed in detail later. I don't know how much time we have left, but I'm going to bring it back to just the idea of this symposium, right? The general purpose is to understand food web dynamics from the San Francisco estuary to the Pacific Ocean. Very lofty goal. We're not going to understand that's not what we're going to do today. We can't understand it in 7 hours. I don't know who that was, but that was funny. But one of the stated goals of this is to think about these processes, these food web dynamics, with the goal of improving ecosystem-based fisheries management, right? So that whether you are a fish person or not, ultimately that is one of the reasons why a lot of the work in this system happens, is to improve conditions for native at-risk species that are listed and protected by the state and federal government, and or organisms that are consumed directly by humans. So there's substantial recreational commercial fisheries for things like halibut, sturgeon, salmon, right? There are many reasons to ask questions about food webs with respect to fisheries management, right? I'm going to posit two. These folks will give you lots more and provide other examples. But, is there something that we can't control, but we need to understand and account for, right? Or is there something we can change? Can we manipulate the system to affect how these food webs work and get some sort of desired outcome? And there's elements of both of this. So habitat, right? We can manipulate habitat. That's the whole point behind habitat restoration, is the idea that it will improve food webs for fishes, right? Tidal energy. Can we manipulate tidal energy? Yes. In some places, in some ways, we can do this, right? Part of it is how we design our habitat restoration, and part of it is creative things like the salinity marsh control gates, right? Like that is control of tidal energy, and it changes food webs. And there are other things that we do that we don't think about in that context. And then flow, right? So in some ways, we can manipulate flow through the estuary. The salinity field rules all, and releases from dams change that. Exports change that. We can't do anything about drought. That's just something that's going to happen. But there are things that we can manipulate, that we can have measurable outcomes that will change something. And then there are things that we just have no control of, but that are super important. For example, there are multi-year lags between changing ocean conditions and ecosystem function in San Francisco Bay, right? And we can't do anything about that. This is, if you haven't read this paper, it's awesome. This is Raymond and Klern, where basically they look at changes in the Spring Transition Index and what that means for the community of fishes and invertebrates in San Francisco Bay. Super fun. And I mean that from the bottom of my heart. Yeah, few just parting thoughts. Ultimately, it is really important for us to understand what levers, knobs, dials, buttons we have available to us and what they do if we're going to be trying to do something about fisheries management, right? But then it's really important, in addition to these things that we can manipulate, we really need to understand what the machine is and how the machine operates. So if you do this lever, this is the outcome, but then also what is the overall ecosystem function like that we're trying to replicate? I had thought I had wanted to discuss like some of my favorite conceptual models in the world. I don't necessarily have time, nor do you want to hear me do it. But mostly I just want to make the point that, scale matters, context matters, variability matters. This is one large system that changes on a relatively consistent spatial gradient, but a wildly variable temporal gradient. And we need to account for all of that as we think about these things and talk about them today. And I'll leave it at that. And thank you for your time.
Let's use the microphone so people on Zoom can hear.
Hi, my name is Ted Flynn from Department of Water Resources. This is mostly a question for Lisa. I was thinking about during your talk when you were talking about restoration and carbon fluxes and things like this. Do you think that considering all the restoration activity that's happening around the Delta in certain areas and the role kind of converting land use change with respect to carbon flux and climate change and things like that, do you think that it seems like we're pretty far from that being part of the regulatory process as it's considered for habitat restoration and how that process plays out? Do you see a path forward in the next, say, 10 years or maybe even longer for that to start to be integrated into the considerations when you're looking at ecosystem restoration and converting land use change for certain benefits like making more tidal wetlands as habitat, in that there might be certain trade-offs when it comes to, yes, we might be restoring it that would create benefits for fish, but it might have negative, larger ecosystem repercussions because it's going to start, cranking out a lot more greenhouse gases or something like that.
Yeah, thanks, Ted. So I've thought about this for a while can you design the best tidal wetland restoration for carbon sequestration, or the best for fisheries, or the best for water quality? What's these sort of, bespoke approaches to what you want out of tidal restoration? I mean, ultimately, from what I am seeing, the just simply tidal reconnection, you guys can correct me, good for fish, good for carbon. I mean, in both ways. So it reestablishes standing water in the soil. It reestablishes dynamics of sediment accumulation and peat growth, because now you're responding to the tides. And there's the one example right now that we have of a freshwater marsh that's been restored is Dutch Slough. And it is, just super productive. It's also making a lot of methane is the issue. But at some point we have to just, I think, step away from the perfect being the enemy of good, and realize that might change as salinity comes in, because one of the big trade-offs, of course, is that sulfate, marine-based sulfate, helps it lets sulfate breathers outcompete methanogens, right? Therefore, yeah, I guess I'm not seeing, I don't like the bespoke restorations where it's got a single goal, because I don't think that's realistic or beneficial in the long run. I mean, ultimately what we should be shooting for is sustainability. So I probably don't have the right answer, but towards that end, we have the data that suggests that historic marshes, I mean, once they start being reached the tidal frame that they need to be, they can sustain themselves, and that is a net sink. So that there's sort of there's no problem with any methane produced at that point, because it's actually smaller at that point, smaller than the amount methane will always be an issue. It just will be offset by the growing carbon in the soil. So I'm not sure if that's exactly your answer, or the question that you had, Ted, but thanks for asking.
Only one more question. Okay.
Great. Good morning. My name is Jay Ziegler. I'm at the State Water Board as the Delta Water Master. And Lisa Marie, Matt, those were great presentations, I think, to tee up discussion today. One of the increasing concerns that I think all stakeholders have across the Delta is the confluence of rising temperatures and water quality conditions that ultimately, at the worst form, manifest as harmful algal blooms and cyanotoxins in the system. And I think neither of you talked a lot about that expressly, but I think I'd just be interested in your thoughts as we kind of start to use and think about water quality indicators across the Delta in terms of achieving both food web goals and protecting other beneficial uses of water at the same time. So I guess and that's a very self-centric question too, because I think that's the lens that I tend to look at issues across the Delta in terms of water quality and protection of multiple public benefits in the use of water. But thank you.
Yeah, so I think there's potentially a lot of things to unpack there. I think what one of the primary things that I see is like a path forward for holistically thinking. I'm going to try to like spin it a little bit to how we're trying to think across systems here. Lisa Marie touched on the MURHAB project, where there's this holistic assessment of harmful algal bloom potential and spread across different habitat boundaries. And I think one of the things that is important to help get people to recognize is that, a harmful algal bloom in South San Francisco Bay or Suisun Bay or Discovery Bay may not be separate or completely isolated from all of the rest of what's going on in the system. So to the extent that we can communicate relatively simple metrics of water quality and this idea of interconnectedness would be great. I don't know if that answers your question necessarily, but just thinking about that. The fisheries related impacts are like a separate thing, and we could talk about that for days.
Yeah, I would just echo that the water temperature question is just the looming issue that I think I'm not gonna say only, but one of the solutions is groundwater, but recharge in that source, but the ocean too, thinking about the ocean as a source of cooling, having a cooling effect as well. And I remember there's more on that. I think John Duran had some thoughts too, but I just want to highlight being connected to the ocean is not necessarily such a bad thing. So I think we have to think about the ocean as a part of the solution space.
Is this on? It is. Bob Nyman, University of Washington, and also a member of the ISAB or ISB. I have a question really out of curiosity, and it has to do with the mass flux of materials. And one of the things that I seldom hear anything about, if at all, is the transport of carbon or nutrients from the greater watershed into the Delta and Bay or through the Golden Gate. Is there a reason for that from a mass balance approach that it would be ignored? Or maybe it's not ignored, I just don't know.
I would not be the right person to ask, but I know someone in the room who thinks about mass balance nutrient fluxes quite often. Brian Bergamaschi from USGS will be one of the speakers in a bit. So he might be able to better answer your question. I do think that's important to think about. It's interesting in the context of, how the rim dams in the system operate. We've talked about it from sediment fluxes, right? A sediment perspective. But in terms of a system-wide nutrient balance, I'm less able.
Only that my understanding is that the rivers are the dominant source of nutrients into the system. Brian, do you have a?
Well, you're talking about carbon transport?
Would have a real influence on the Delta and Bay. But as a bit of a kind of a scientific outsider to the Delta, it seldom comes up in conversation. It seems as though we have been legislated to talk about certain things that go on in the Delta or the Bay, but nobody seems to talk about the larger landscape very often and what's going on there.
Well, you put your finger on something that's bugged me for approximately 30 years here, in that we know we've got a massively heterotrophic estuary, but we don't try to quantify all the carbon, including the terrestrial carbon inputs and the detrital input, the non-algal components that are coming in from the river systems, et cetera. I think it's because it's easy to measure the phytoplankton, so we focus on that. So it's like the guy, looking for his wallet under the street lamp. We can measure that, and certainly it's important, and certainly that kind of carbon is important to the food web. We are just concluding a study where we're trying to suss out the origins of and the different food web benefits of different sources of carbon across the system. So it's a huge hole, and I think, in what we know about the system.
Okay, so with that, we close this session. Thank you so much, the keynote speakers. And we will have a very short break. We should restart with the introduction of the next session at 10:10. So it's 10:04, but 10:15 is the talk. Yeah, we don't want to run late. So in 6 minutes, so some coffee, bathroom break, things like that. The introduction. Let's take our seats. We will introduce the next session.
Grab your seat. Okay, all yours. All right, hi everyone. Gonna let you all get seated. I just want to point out that I am, unlike Matt, not overly caffeinated. I'm suffering from crushing sleep deprivation from a 1-year-old who's getting her front teeth. Anyways, no amount of coffee can help me right now. I'm really excited to introduce the speakers for this session. This is the modeling session. And we are going to be shepherded through complex model frameworks that use multivariate methods such as structural equation modeling, or SEM, by Sam Beshevkin, and multivariate autoregressive state space modeling, or MARS models, which I will be presenting on. And these methods leverage multiple disparate sources of data to shed light on long-standing questions about forage fishes, which are often considered a critical link in the pelagic food web. So our first speaker is me. So I'm Denise Colombano. I'm a senior environmental scientist at the Delta Science program. You'll get to know me and my love of synthesis and fish during my talk, so I won't dwell on myself. Then I'll be followed by Dr. Sam Beshevkin. Sam is an environmental program manager at the California State Water Resources Control Board, where he works on efforts to update and inform implementation of the Bay Delta Water Quality Control Plan to establish flow requirements for the watershed. But he is widely known in the Delta science community for his open science activities and cutting-edge statistical analyses of long-term data sets which span water quality, zooplankton, fishes, and monitoring survey design. Raise your hand if you've ever used one of Sam's packages or data products. Yeah, if you haven't, check them out. Our speaker after that is Dr. Bob Nyman. Throughout his research career, Dr. Nyman has studied the structure and dynamics of riverine ecosystems, including riparian vegetation and the role of large animals in influencing ecosystem dynamics. He is the author of 11 books on aquatic ecology and watershed management and over 230 journal articles. He has served as director of the Matamek Research Program of the Woods Hole Oceanographic Institution, director of the Center for Streamside Studies at the University of Washington, a visiting scientist on numerous occasions with the Centre National de la Recherche Scientifique. All my French classes really worked for me there. In Toulouse, France, and a visiting professor at the University of the Witwatersrand in Johannesburg, South Africa. He's also been a professor at the Center of Excellence in Natural Resource Management at the University of Western Australia. We are so honored and lucky to have you. But I'm not going to stop there. He's also earned numerous awards for his academic contributions, including, I think this is so cool, the Eminent Scientist Award from the Ecological Society of America. So he currently serves as a member of the Delta Independent Science Advisory Board, which you will hear being referred to as the Delta ISB. Okay, so those are our speakers for the session. Now I'm going to start with my talk. So today I'll be presenting on a modeling effort that is about Pacific herring population stability across the San Francisco estuary. And I will highlight my authors, but Jim Hobbs is here in the room. So thanks, Jim, for helping make this project happen. And I want to start off with a quote. This is a real inspirational quote for me, published over 10 years ago in this article, Big Data and the Future of Ecology. Our capacity to efficiently provide timely information to meet modern societal challenges will depend on a global greening of ecology. That is, data should not only be generated and analyzed, but must also be available to be reused and recycled. And I read that 10 years ago and I really see it happening today. So I just find that very inspiring. What the authors of that paper are really invoking is open science. And open science is a global movement that promotes transparent scholarship at every stage of the research life cycle. So here in California, we have AB 1755, which mandates these practices for water and ecological and fisheries data. This is to support synthesis science across data sets and disciplines, which Lisa Marie highlighted, and to help water managers operate more efficiently. But what is synthesis? Synthesis is the process of combining disparate sources of information to see the bigger picture and gain new insights. And synthesis at the Delta Science program comes in many forms. We analyze and synthesize scientific information to report on the status and trends of key Delta issues. We update the scientific state of knowledge, identify needs and data gaps, and guide adaptive management. So synthesis can be found in the Delta Plan, the Delta Science Plan, the Science Action Agenda, and the State of the Bay Delta Science collection of articles. If you're interested in finding our open data products, they're hosted on our website. For example, Shiny Apps, GitHub repositories of tools, data sets, and project workflows, and the Delta Science Tracker, which is a brand new web platform that tracks and summarizes scientific studies in the Delta. But to move on to this study, I'm going to be presenting on an open science synthesis project. This publication came from a collaboration between UC Berkeley, where I was a postdoc, and the California Department of Fish and Wildlife. It's funded by a California Sea Grant and a Delta Science Fellowship. Nina Pack and I co-led the project. Stephanie Carlson and Albert Ruhi were the PIs. Tom Greiner from the Marine Region and Jim Hobbs from the Bay Delta office provided expert on-the-ground knowledge of the Pacific herring fishery. So for those of you who don't know, the Pacific herring is a fairly long-lived forage fish that can spawn multiple times in its life. And this is unlike salmon, which spawn and then die. Adults living in the ocean move into the estuary to spawn on shorelines with structure. So anything, eelgrass meadows, rocks, pier pilings, even on boat holes. The eggs incubate for several weeks, and then the larvae are either retained in nearby habitats or they drift around as plankton. They spend their first year rearing in the estuary and are often found in shallow embayments and tidal marshes. Then they move out to the ocean and recruit to the adult population and start the cycle all over again. Now, herring is a classic mid-trophic level planktivorous forage fish, serving as an important prey item for many upper trophic level predators in the ecosystem. Both herring and their eggs are an important food resource for many large fish, seabirds, and marine mammals. So here's an inventory of 59 known predators of herring in the California Current. Pretty impressive. But it's not just that they're consumed by many predators, they comprise a large proportion of predator diets. For example, this is a quote from a paper, the diet proportion of herring for salmon, humpback whales, and murres was between 15 and 30% on average in the Gulf of Farallones for the winter half of the year, from October to April. So in fact, when years where large numbers of spawning herring came into the estuary, you also see that reflected in Chinook salmon diets in the ocean. Perhaps lesser appreciated, but equally, if not more interesting to me, is their role in the estuarine food web, as demonstrated by these two quotes. And this is from Rosie's paper. Of the 464 larval fish found in Delta smelt stomachs, 52% were Pacific herring. 8% were prickly sculpin, 1% were longfin smelt. 1% were Tridentature species, and 38% were unidentified. So this is from a Delta smelt study. The second one, a few uncommon prey observed in the diets may hint at feeding strategies employed by the larvae, such as herring DNA in the longfin smelt diets, suggesting feeding on eggs near substrates. So this is from a longfin smelt eDNA study. So collectively, based on these two findings, it means that herring are also a food source for pretty much everything in the ocean, but also for endangered smelts in the estuary, which I find totally fascinating. And hope you do too. But they're not just a food source for other critters, they're also an important food source for humans. They support commercial, recreational, and subsistence fisheries in San Francisco Bay. These are pictures of the sac roe, which is a delicacy, whole fish, which is comprised of a small fish market, and then eggs on kelp, which is a subsistence fishing method. But unfortunately, record low numbers of spawners starting around 2008 caused emergency closures of the fishery, prompting managers to look more closely at drivers of population stability. So what is population stability? Well, if we look over a 40-year time series, Pacific herring often follow boom and bust cycles. So you can see them go up, you can see them go down, you can see them go up again. However, managers noticed that the adult spawner biomass index was not only declining over time, but was also becoming more variable, more erratic. Looking more closely, the first 20 years were stable. The second 20 years were unstable, with multiple fishery closures as shown by these arrows. In response to these fishery closures, the department reevaluated their approach and developed a model to forecast adult spawners. So here's a model by Bill Sidman that's used by the department in their new fishery management plan. And basically, the model is, if the forecast falls below a certain threshold, they proactively close the fishery for the upcoming season before it begins. And this is the model. Adults entering the estuary to spawn in wintertime is a function of the same metric 1 year prior, plus ocean temperatures during the summer and fall leading up to the spawning season, and the number of juveniles rearing in the estuary 3 years prior. So in our study, we asked the question, if the number of juveniles rearing in the estuary 3 years prior is a predictor of adults, what predicts the number of juveniles? So we hypothesized that a number of factors could be important. There's rearing and spawning habitat loss. The estuary has lost a lot of its natural shoreline habitats like eelgrass meadows and tidal marsh. There's upwelling frequency and duration, as Matt alluded to. The spring transition index has shifted its timing and later and become shorter. Marine heat wave events. Maybe you've heard of the ridiculously resilient ridge, the blob. It's making anomalous heat waves more frequent, but more persistent. And then there's flood and drought events, which we have been referring to as weather whiplash, making our precipitation regimes more extreme. And there's also the number of adult spawners. If they get low, maybe they're making less eggs. So you may have seen this in the news back in 2019, and there was a lot of speculation about global warming. So we started by looking at regional time series of salinity and temperature in the estuary. So basically the rows are regions and the columns are salinity and temperature. If I can figure out this laser pointer. Ah, yes. Okay, so they show an estuary that's highly dynamic, that shifts from fresh to salty and back again, and then cool and warm and back again, and so on. But it's also an estuary that's recently experienced more extremes. So for example, the record drought starting in 2013 and the record heat wave starting in 2014 co-occurred. So based on these hypotheses and the available data sets, we developed a conceptual model of what we think influences the number of juveniles rearing in the estuary in spring. And in this model, the number of juveniles is a function of the adult spawner biomass from the prior winter, plus temperatures in the estuary, plus salinities in the estuary, plus competition for resources, such as food and cover, due to habitat loss or resource scarcity. And we're going to refer to this as density dependence. The two different data sets are from the California Department of Fish and Wildlife's San Francisco Bay study, and then also the egg deposition and spawner survey. And since this is in the modeling section, I'm going to briefly show the Mars model structure. I'm happy to explain this in more detail to anyone interested. I think in the interest of time, I may gloss over it, but basically there are two equations within this, and you use one equation to take the observations and then estimate the true abundances. And one thing that I want you to take away from this is that in our model structure, we were able to estimate density dependence, which becomes important later on. So in terms of key findings, our first key finding was that the top rank model had 3 out of 4 model terms. That's weird. Okay. Well, anyways, I can just talk about them. Temperatures in the estuary, salinities in the estuary, and competition for resources. Specifically, the model showed that juvenile herring were generally associated with cooler water and saltier water in the estuary, and that they were strongly density dependent. That is to say that a scarcity of resources likely caused competition and mortality, limiting their population growth. Our second key finding was that the Central and San Pablo Bays were major contributors to the population. The estuary-wide population as a whole was 15% more stable than in its individual regions, meaning that unique regional variation in these population dynamics, in their habitat and their food availability, conferred stability to the estuary-wide population. To recap, we built a conceptual model and tested our hypotheses with 2 existing data sets to uncover drivers of the number of juveniles rearing in the estuary, which we know is a key predictor of adult spawners 3 years later, according to this forecasting model. So what does this all mean from a management perspective? It supports the idea that boosting juvenile production in the estuary may help prevent fishery closures. To boost juvenile production, a promising area to focus is improving rearing conditions via habitat restoration. So habitat restoration will look different in different regions, but in particular, we emphasize the importance of eelgrass meadows and tidal marshes. They're productive vegetated habitats that are great places for juveniles to eat and hide from predators. And the good news is that this is very consistent with a number of existing restoration initiatives in the Bay Delta. Bad news is that, unfortunately, marine heat waves are a major, major issue for both juvenile and adult life stages. So mitigating the effects of marine heat waves by providing refugia is a topic that needs much more attention from scientists and managers. That was one of the messages of our paper. So if you're interested in reading this paper and accessing its data products, it's totally open. There's no paywall. You can look at the code. You can look at where the data came from. Feel free to scan this code or email me and I'll send you the links. In the spirit of open science, I'm going to end with one more inspirational quote because I want to leave you on a happier note than marine heat wave effects. Okay. Our ability to produce specific analytical information for local problems that can also address questions at larger spatial scales and over longer timeframes depends on our willingness to work collaboratively to collect, preserve, and share our data across projects, locations, and research groups. Okay, thank you for having me.
We have time for a couple of questions.
I have many questions, and I think I'm going to save. I have a deeper question about the role of forage fishes and ecology of forage fishes in the Bay Delta Ocean continuum. But so just as a teaser, that is going to come later. But why no herring in the South Bay?
We had herring in the South Bay. I don't remember. Oh, sorry, I went past it. Here. So the bottom left panel is South Bay. So there, when you say no, it was the third ranked contributor to the overall population. So it was actually pretty stable and had like a stable but minor contribution to the overall population.
Do you think that that changes as salt pond restoration matures and habitat in the South Bay changes over time, or do you think that it's sort of off the axis and isn't just a huge contributor?
I think that's a Jim question, honestly, since seeing as how he spent his entire life in the South Bay looking at fish. Jim, do you?
Certainly the larvae in early life stages are using those tidal marsh restored habitats in South Bay. But we're not seeing a lot of spawning in the South Bay. So it's mostly just sort of larval drift from the North Bay. So it's kind of it may be somewhat of a sink actually to Pacific herring recruitment in the South Bay.
All right, this is a typically hard question for a model. So your model, you start out with those 4 components. Can you just comment on all the other factors that were not in your model?
Of course.
That might also like have been contributing, and that being like just such a major challenge for trying to figure out controls on food webs and targeted organisms.
Yeah, absolutely. There are so many factors that could be affecting Pacific herring. And so it was tough when we were looking for available data sets to really figure out like what we could include in the model. So for example, we would have loved to include zooplankton data directly as, to model their prey source. And the way that the Bay study works, it's sampled monthly along this transect from the South Bay all the way up into the Delta on a monthly time series from 1980. And the zooplankton data that would be in the same vicinity doesn't start till 1995. And I believe that it only goes down into San Pablo Bay. So you have a mismatch in time and space of overlapping data sets. What we realized is that marine productivity and upwelling is correlated to the temperature in the bay, because we have these plots of temperature here, and actually based on the paper that Matt showed, Ramno and Klern, they show that there's synchronous fluctuations in temperature at the Gulf of Farallones and in the bay. And then as you go inland, it kind of gets decoupled. But temperature for us sort of approximates, conditions that would be good for herring in terms of food supply. So it's sort of an indirect measure. But then the other thing was the density dependence. It gives us insight into like limitations on population growth. And then we can make inferences based on that. Well, it could be the availability of food, could be the availability of cover. We can't say for sure. We're looking forward to more studies on herring, like from Jim's work and other studies around the bay to actually quantify that. But that's sort of our way of like kind of trying to get at it. But certainly it would have been nice to have more fine scale resolution data on that.
Okay, so let's thank the speaker, and we have next is Sam.
Hi, everyone. Thanks for having me. So I'm Sam Bashevkin with the State Water Board. This work that I'm talking about was conducted mostly during my prior position when I was actually at the Delta Science Program. And it was co-led by myself and Tanya Rogers with the National Marine Fisheries Service. And we had a bunch of collaborators that you can see on the screen, and many of them are in the room here today, which is great. So I'll be talking about our work to identify drivers of food web dynamics. So what drives food supply on an estuary scale? The key issue we're thinking about is that key fish species are declining, which is linked in part to declining food. But we have a limited understanding of the drivers of the aquatic food supply. Why is this important? Because understanding food supply drivers will inform effective management actions that provide optimal conditions for the recovery of declining species. So just realizing that hopefully most of us understand why food webs are important, I'm just going to jump right into the study. So the main questions they were asking are, first, what is the relative influence of abiotic drivers, bottom-up effects, and top-down effects on different components of the Delta food web? We focused mostly on phytoplankton, zooplankton, and fishes. And how do these effects vary among different regions of the estuary and between annual and monthly timescales? So to answer big questions like this, you need to integrate data. And we integrated nutrient and chlorophyll data, flow data, clam data, zooplankton data, and fish data. We computed missing data at the station level for nutrients, water quality, and chlorophyll using ARIMA models. And these data sets were all then combined into annual and monthly data sets within the 4 regions you can see on the map on the slide. And note the 4 regions on the map since they will be used later on when we describe our results. The upstream extent of our study was limited to the regional boundaries you can see on the map here because of the lack of data outside these areas in earlier years for all of the variables that we were looking at. The analytical method that we used was structural equation modeling, or SEM, also known as path analysis. So this method is very similar to multiple regression, but it allows for multiple response variables that are linked together in a network, expressing some causal hypotheses about how the variables interact. Using this approach, we can model both direct and indirect effects, as well as residual covariance and latent variables. And a latent variable is a model of some underlying state, for example, fish biomass, which is manifested with error by multiple indices, for instance, different surveys of fish biomass. Our analytical approach was to first develop a conceptual model of the Delta food web as a group, and this was based on known trophic relationships and the natural history of the system. We then simplified the model to what was actually feasible to fit given the data we had, which involved using aggregates or different subsets of variables. We then fit the model and evaluated model fit. In the event of poor fit, we made modifications to the model, not in an arbitrary way, but in a way that was consistent with our understanding of the ecology. And when that was completed, we attempted to make some sense of the very many results that came out. This was the conceptual model of the system that we developed, which was based on the group's collective knowledge of the system, but also somewhat by data availability. So this expresses the relationship between nutrients, phytoplankton, for which we used chlorophyll as a proxy, various zooplankton groups, clams, estuarine forage fishes, which were an aggregation of these 5 species, their predators and competitors, and 3 abiotic drivers that we settled on: flow, temperature, and turbidity. We considered other abiotic drivers, but excluded them due to high collinearity, for example, between flow and salinity. We used biomass per unit effort for all biological variables except the clams, for which only count per unit effort was available for the full time series. And we developed models at 3 different levels of spatial and temporal organization, which were paired with the annual and monthly datasets that we developed. For the annual model, we used trophic level aggregates for zooplankton and a latent variable for fish biomass that was manifested by 3 different survey indices. Due to time constraints, I won't be discussing the annual model today. For the monthly model, we divided our analysis into 3 submodels, one using upper trophic level aggregates, specifically zooplankton and fish, another using lower trophic level aggregates, specifically nutrients and phytoplankton, plus clams, and one that used the individual zooplankton groups. Since many of the monthly variables were highly seasonal, we ended up removing the seasonality from all variables by subtracting the monthly means, so that we were essentially modeling the relationship between seasonal anomalies. We also used 1 month time lags as predictors, which allowed us to simultaneously estimate bottom-up and top-down effects, as well as the effect of self-regulation or autocorrelation. Also, due to time constraints, I'll only be presenting today on the upper and lower trophic level models. So this was our upper trophic level model at the monthly time scale, which had 3 response variables you can see in the middle column. So these were estuarine forage fishes, specifically the index from the base study midwater trawl, as this was the only survey to sample year-round. Predatory zooplankton, which were the sum of mysids and predatory copepods, and herbivorous zooplankton, which were the sum of cladocerans, herbivorous copepods, and rotifers. Phytoplankton, represented by chlorophyll, was included as a bottom-up driver, and clams were included as a top-down driver, where the species of clams used in the model depended on region, since different species are found in different regions. Top-down effects on fish included age 1+, striped bass, centaurochids, marine fishes, and silversides, and these variables depended on region. Turbidity here and in all of our other models was represented by negative Secchi depth, which we did just to aid in interpretation. And so in this diagram, the different colored arrows represent the different types of interactions represented by each path. To make these results more digestible, I'm going to present each interaction type sequentially. The numbers you see here under each of the response variables in the middle column are the R-squared values for each response variable. So as you can see, the model doesn't explain a majority of variation in any of these variables. So you should take these results with the appropriate grains of salt. In the following figures, blue arrows will represent positive effects and red arrows negative effects. Gray arrows will be non-significant effects, as you can see in the key in the upper right corner. And you'll notice there's a separate plot for each region. So first, starting with self-regulation, we found that most everything is a good predictor of itself the following month, which is not very surprising. For bottom-up effects, we generally see positive effects of lower trophic levels on higher trophic levels, and these effects seem to be more prevalent in the Sacramento and San Joaquin regions than in Suisun and San Pablo. In contrast, top-down effects tended to be more prevalent in San Pablo and Suisun than in Sacramento and San Joaquin. We can also see that the Potamacorbula clam species has a negative effect on zooplankton, whereas the Corbicula clam species in Sacramento and San Joaquin does not. And this is consistent with Potamacorbula being known to have a higher grazing rate. And also top-down effects on fish only appeared in Suisun, and they were unexpectedly positive. The effects of abiotic drivers were various, and I don't have time to discuss all of them, but I will point out that in 3 of the 4 regions, higher turbidity was found to have positive effects on fish. And then this is what all the results look like together, where the colored arrows are the significant paths and the gray arrows were paths that were not found to be significant. So moving on now to the lower trophic level model. This model also had 3 response variables, which are clams, phytoplankton represented by chlorophyll, and nutrients, specifically dissolved inorganic nitrogen, or DIN. DIN was a combination of ammonia and nitrate, which we used because nitrogen is thought to be more limiting in the system than phosphorus, and also because many of the nutrients were correlated with each other. In this model, the effect of phytoplankton on nutrients was not lagged because the lag wasn't supported by the data. This model also included effects of higher trophic levels on subsequent nitrogen levels, which can be interpreted as nutrient cycling. We have the same setup here for the results. I'll point out that the R-squared value for phytoplankton was very low in all of these models, suggesting we don't have a very good handle on drivers of chlorophyll in this system, at least with the variables that we used in our models. So as before, all variables showed a positive relationship with past values. Bottom-up effects, in this case, were only present in San Pablo and Sacramento, although the sign of these effects was not as we would always expect. In terms of top-down effects, high phytoplankton leads to reduced nitrogen in all regions, which some recent research suggests could be due to rapid nutrient uptake during phytoplankton blooms. And abiotic effects, again, were various, but I will point out that the effect of flow on nutrients was negative in all regions except San Joaquin. So this model is maybe capturing the dilution of wastewater inputs along the Sacramento corridor. And finally, we found one case where upper levels have a positive effect on nutrients in the San Joaquin region, so that nutrient cycling. And this is what that lower trophic model looks like altogether. So if we look at the total effect size for each type of interaction, which you can see as the rows here, for each of the 6 response variables in each of the 4 regions, and color the ones that are statistically significant, we get this table that you're looking at now. Some summary points are that self-regulation is important to consider for many species on a monthly timescale, that the relevance of bottom-up and top-down effects varies by taxon and region. For instance, for herbivorous zooplankton, top-down effects are more prevalent in the outer estuary, but bottom-up effects are more prevalent in the inner estuary. And also, abiotic drivers were important for many species, although the effect of any particular driver will vary by region and taxon. Some considerations when interpreting the results. So as I mentioned earlier, many variables such as chlorophyll had a lot of unexplained variation. Also, the data in some cases suggested paths that appear to jump trophic levels. These were paths that were added during the model evaluation and modification phase to properly reflect covariance in the data. Also, some of the signs of the paths were not as expected based on a priori knowledge. There are several possible explanations for these results. These include missing variables or missing indirect effects, as well as interactions among predictors, so non-additive effects and non-linear effects, neither of which are accounted for in this modeling framework. Also note that while we found that bottom-up effects seem to be more important for fish than top-down effects, we lack good data on large predators of fish. Also, we ended up excluding many abiotic drivers due to collinearity, so differentiating among them and determining which drivers are most important does remain a challenge. Nonetheless, our findings can suggest interactions that are supported by the existing integrated data, which could be used in the development of other types of food web models. So in conclusion, we found that evidence for bottom-up drivers of food web dynamics supporting food supply as a critical management objective in the estuary. We found positive bottom-up effects of chlorophyll on zooplankton and of zooplankton on forage fishes. We found that the net effect of environmental drivers was typically on par with or greater than bottom-up and top-down effects. In particular, flow and turbidity were important. For zooplankton and estuarine fishes, bottom-up effects were stronger in the freshwater upstream regions, the lower Sacramento and San Joaquin rivers, while top-down effects were stronger in the brackish downstream regions, Suisun and San Pablo bays. Lastly, we really need better monitoring at the top and bottom of the food web. Like I just mentioned, we lacked good data on the predators of these fishes. Most of the fish monitoring is focused on smaller fishes rather than the larger fish predators. And as we saw from the low R-squared for chlorophyll, we really lack good data at the very bottom of the food web as well. So those are 2 areas maybe to improve our monitoring enterprise. So lastly, I'd like to thank the Delta Science Program and NCEAS for facilitating this working group, as well as the instructors and organizers who assisted us along the way. And also I'd like to thank all of the IEP monitoring programs that collected this data and the survey staff who are out there doing it, without which none of this would be possible. Thank you.
Have
time for one or two questions.
Thank you for your talk. My name is Sophia. I'm a PhD candidate here, and I have two questions. One is if you could speak a little more to the decision to focus on those 4 regions, and that being like the scale that you all decided was the best to make a comparison. And then the other is a little bit more technical. I noticed that you said the in the upper trophic level model, in most of the regions, higher turbidity had a positive effect on fish. And I was wondering if you had a hypothesis as to why that would be, if you looked more into that.
Yeah, thank you. So for the first question, the regions, I mean, that was really just the only option based on the overlapping data availability. So I went back to the slide showing all the variables that we're using, and we could only model instances where we had data for every single one of these variables. And it was just a factor of where they happened to spatially overlap. And that was the region you can see outlined in the map, depending on the time scale that we use. So for the monthly models, I think it was from 1980 to roughly 2020 or whenever the most recent year available was. So, going back that far, this was the only region where we had data for all of those variables. And we wanted to look at, regional divisions because of the there's a lot of studies on the salinity gradient. They're, very different habitats as you move throughout there. So, we're trying to capture kind of the main regions that are, San Pablo, then Carquinez Strait is kind of a border. Suisun has some unique dynamics, and the lower Sacramento and San Joaquin rivers before the confluence have, unique divisions. So we're just trying to recognize that. We did get reviewer comments suggesting that we should model it based on salinity rather than based on geographic regions. And we looked into that. I mean, I think we acknowledge that that would have been a better approach, but given that the monitoring stations are geographically fixed, when we actually looked at what it would take to have stations lining up based on salinity bins rather than geographic regions, there would have been so many gaps in the time series that we wouldn't have been able to monitor it. So, if we'd gone back in time to 1980 and told the monitoring programs to have every single station based on a fixed salinity level, then we could have done that type of model. But without that, or without using more advanced modeling techniques, the geographic regions are really the only option that would work for us. For the other question about turbidity having positive effects on fish, I think there's a lot of literature on the positive effects of turbidity. I mean, Matt is probably better equipped to talk about that than me. My understanding is that, for Delta smelt in particular, it's kind of 2 benefits. One is providing them refuge from predators, and second, in facilitating their prey themselves on zooplankton. But that's about the extent to which I know. I mean, the San Francisco estuary, I think, is known as a high turbidity estuary, and it's recognized that many of the species have adapted to that. But yeah, I'd encourage you to maybe ask Matt about that. I think he would know a lot more, or the other fish experts in the room. But thank you.
Okay, so let's thank the speaker. We have to move to the next one. And please remember that any lingering questions, please put them in the system we have. We will get to them in the panel.
Would you please?
I just assume use this one.
That pop button, it takes a sec.
Yeah.
There's like.
Okay, thanks, Denise. Okay. Thanks so much for inviting me. I really appreciate it. Well, there's lots to talk about today and great presentations so far. Where to begin? Well, I guess I should begin at the beginning here. First of all, I'm going to, in the next few minutes, talk about a report that's still evolving. I'm going to tell you about some things that, that we internally have talked about and are putting out there for public consumption, but we also appreciate your feedback. And there's still time, to get your thoughts in as we revise the report over the next several months. So with that said, just a little bit of preamble here about who we are for those of you in the room who are online who don't know that. The Delta ISB is made up of 10 scientists from across the country, and we have various disciplines on the board. We're mandated by California legislation to provide oversight of the scientific research, monitoring, and adaptive management that's going on within the Delta. And we usually do this through preparing reports on various topics from time to time. Now, the report I'm going to talk about today, the review, has to do, of course, with food webs. And this too is evolving a bit from our initial discussions that we had well over a year or so ago. But what it's come down to is to really assess how to better incorporate food web knowledge in managing the Delta's ecosystems and to identify what tools might be appropriate for really advancing that understanding. Now, also our perspective has evolved over time too. Initially, we were really thinking about with having a focus just on the upper trophic levels and how these top-down effects might be influencing the Delta ecosystem. But over time, we've come to really consider in the report the full food web. And I think you'll see that in what I'm going to talk about over the next few minutes. So how we did the review was, I guess, normal standard procedure for the Delta ISAB or ISB is we evaluated the existing literature such as we could find it. Granted, there were lots of publications out there, especially in the gray literature, which we're still trying to assimilate into the process. We did have extensive discussions with the Delta community over time with many of you in this room and online. We held a 2-day workshop where we had over 100 participants putting forth their ideas. And we're in a public comment period now. And we're receiving some actually just excellent suggestions from the scientific community as well as the public on how the report, might be modified or edited or other items that might be included. So in general, what did we find or what have we found so far? On the right, you'll see that there are several food web models already developed for the Bay Delta. These are just examples. Many are focused on the role of bottom-up processes in structuring the food webs. Not so many until recently with top-down. Many of the Delta models that attempt to present entire food webs are really limited in their spatial and temporal coverage or are conceptual in nature. And that's probably to be expected in any case. And everybody we've spoken to so far, virtually everybody we've spoken to, have said that any effective evaluation of the management or species population changes really requires a quantitative approach. And that seems to be on everyone's radar as to how to move ahead. Well, as part of the process, we tried to identify what would be the benefits of adopting a food web perspective, a food web approach, or a food web strategy for the Delta. And we examined 5 possible applications for improving how this might improve natural resource management in the region. And I just have one example here, and that has to do with ecosystem-based management. But we also considered restoration actions, environmental change, climate change, the role of contaminants, and so forth. And for each one of these, we put forth a management question. We identified the benefits or possible benefits of a food web approach. And then we offered some suggestions for what those priorities might be. And I do have to admit that these are evolving, continuing to evolve as we go through the public comment period. for example, with the ecosystem-based management, how do changes to environmental conditions affect food web interactions? Well, we've received several suggestions of modifying this particular question or how we present this. So I'm not going to go through this in great detail right now. You can read it, but that's been our approach. We also tried to bring in, or we brought in, people from other systems from around the country, 5 in particular, ranging from the Gulf of Mexico, Chesapeake Bay, the North Atlantic, and so forth, where they have adopted a food web approach and tried to show, at least the participants in the workshop, as we do in the report, of how these programs are benefiting from taking a food web perspective in managing their fisheries and in managing their environmental conditions. So as part of the process as well, there were a number of science gaps, or what I prefer to call them as challenges, that were identified. And I'm going to give you just 7 here very quickly. The first one has to do really with linkage between the primary producers and the upper trophic levels. Now, a lot of work has been done in the Delta on this, and as well as in San Francisco Bay. But there, as we'll see probably in the following presentations, there's still a lot that needs to be done here. The second has to do with better characterizing the role of benthic microbes and invertebrates in food webs. Now, we realize that there's a lot of ongoing research in this area, in certain areas in the Delta, very active programs at the moment, and we would like to highlight these in the report and I guess offer encouragement for the efforts that are being made today. One topic that came up over and over in our discussions was that the role of detritus seems to be very poorly understood in terms of how it might affect the carrying capacity and overall productivity of the Delta. It seems to be a topic that has not been deeply researched in this area, but deserves and is recognized as being deserving of a lot more investigation in the coming years. The fourth topic or challenge that came forward has to do with the role of the most abundant fish species in the Delta. We seem to know a great deal about salmon, smelt, bass, and so forth, but there's many other fish species out there and relatively little information on their abundances that we can plug into the models of interest for us to really understand the roles that they play in terms of top-down controls. Almost everybody identified the role of contaminants, even sublethal effects of contaminants, as being a great unknown in the system, and that is echoed in the report. The quality and consistency of food web data for synthesis. This is a great concern, or not so much the food web data itself, because there's a lot of ongoing efforts at the moment to really build up the databases on food webs. But it has to do with syntheses. We have a large number of people with a lot of experience in the Delta that are retiring over the next few years. And with them goes a lot of knowledge about the Delta. And we would, from the Delta ISB's point of view, we would like to see not only the data sets, be available for other scientists, but also the knowledge of these folks that are retiring captured in some good way, and so that we produce syntheses of our knowledge going forward. And that's been echoed several times in the previous presentations, and will be, I'm sure, in the future. And then finally, we feel in terms of a challenge, there's a real need to evaluate the role of predatory birds and mammals, and in this case, including humans as one of those mammals, of human consumption, harvest, whether it's recreational or commercial, of how that's actually impacting the Delta's food webs. I was very surprised to learn that how little was actually known about the role of birds and other mammals other than humans on the system. So what are our tentative recommendations and what might be a way forward here? Well, I'm going to throw these out now and take them as tentative. You know, we're moving in this direction, but we're still listening to the community of what you think the recommendations might be so that we make them stronger and more useful for decision makers. The first one is to really develop a comprehensive coordination and implementation plan for collecting, analyzing, and applying the food web information. Now, whether this is done through focused interdisciplinary collaborations, like we currently have some going on through the Interagency Ecological Program, but probably more is needed on a broader scale. We also feel, the Delta ISB also feels that it should be you as a community which decides on what the key management needs and science questions might be going forward. We can offer some suggestions, but it's the Delta Bay community that needs to decide. And then finally, the activities identified by the plan should be initiated using hypotheses and, where possible, established testable experiments and establish as testable experiments, excuse me. And this is being done in other places around the countries, such as in Chesapeake Bay and elsewhere to help guide their programs. The second recommendation that we have is to adapt the Delta research and monitoring programs to explore some additional key aspects of food webs. We suggest that there be a greater focus or emphasis on the science gaps, and then as well as connecting the existing knowledge. There's a huge and very viable knowledge base within the system. It's unbelievable how much knowledge is available here to many of us who have worked in other parts of the world. Use it. It's actually something that is one of the strengths here, if it can just somehow be harnessed and pulled together. The food web data within the system need to be regularly updated, quality controlled and made accessible. And we're starting to see that now through the IEP programs. And then encourage and support ongoing syntheses of existing food web information. This is something in all of our discussions that does not seem to be done except on very specific topics. But having, you know, kind of some of the senior people in the Delta having the ability and the time and encouraged by their agencies to do this would be a real step forward. So data management, information sharing, and synthesis, we feel, are pillars of a well-functioning science management program. You know, and I hope everyone will take this to heart. Our third recommendation has to do with employing appropriately scaled food web models as determined by whatever management questions you decide on and the environmental driving forces. And I put up a couple of examples here, one from John Duran and the other from Andre Buchheiser, of how they've used these models, or how they've developed processes for actually developing the models further. And we encourage everybody to look into this much deeper and use the process of model development that they have followed. And then finally, it's adaptive management. And it's something that, again, I'm surprised from time to time that even though we talk about adaptive management over and over again, that in many of the proposals received by the Delta Science Program and elsewhere, adaptive management is not included. It's getting better, but it's something that needs to be really at the heart of many of the programs here, or all of the programs wherever possible. So these are the take-home messages out of the report. We feel that advancing food web science in the Delta will serve a broad range of management applications. And we also feel it will improve the ability to forecast the effects of management actions and climate change on fish and other aquatic species. And the collaboration and adaptive management will increase the effectiveness and efficiency of the programs that are ongoing right now. Then the Delta ISB fully realizes that these recommendations that we're putting forward for public comment, that these are really ambitious. We're talking about almost like a sea change in the perspective or strategy of how science is done within the Delta. And these will take time to be developed. I don't see this happening next year or the year after. I see it happening probably over the next decade. It's going to take time, especially as I think a lot of the younger scientists move into positions of making more decisions or having more control over their programs. The Delta ISAB or ISB also believes it will require multiple workshops and team building activities. We went through our process. It's now time for the community to go through theirs. And this, we would see one or many actually follow-up workshops on food webs, going forward. And that this is a whole community effort, just like this example that you see on the right, which is for Chesapeake Bay, which literally includes everybody, from decision makers down to technicians and the public on what goes on in Chesapeake Bay. And the bottom line is the fate of the recommendations that we put forward in this report are going to largely depend on whether the Delta community deems them to be sufficiently important or not. It's really up to the community, once this report is out. And anyway, I say thank you to everybody for listening. The report is still available for public comments. You can use the QR code here to download the report, or if you need a hard copy to look at, there's actually a number of hard copies at the registration table as you come in. And we really encourage you, we encourage everybody. We've received excellent comments so far, really thoughtful comments from a variety of folks within the Delta. And we hope to hear from more. And comments, we will accept comments up to June 14th. And then it's sort of the ball's back in my court and the rest of the Delta ISB to pull this together. And hopefully we'll have it out as an official report by the end of the summer. That's my wish. Anyway, thank you very much.
Time for one or two questions.
So is the ISB considering, ambitious recommendations, fantastic, ambitious recommendations, aspirational, true aspirations that can only be achieved if there's resources available to achieve them. So is part of the consideration of the ISB how the next generation of scientists will be able to gain access to the resources to realize your vision?
Yes, thanks for that question. That's a good one. It's one that we have discussed internally, within our group, and we fully understand that. We see this report acting as a catalyst to leverage those kinds of resources, and wherever possible, we're more than willing to help to try and move whoever makes those decisions about resources, move them towards a food web perspective so that those, become available. I think if the report is done right, like many reports, most reports sit on a shelf. As you know, you've been around long enough to know that the great recommendations go to the shelf. We're trying to make this report such that it acts as a true catalyst to actually move the science enterprise within the Delta and the Bay, closer towards a food web perspective. And that means, being able to redirect some of the existing resources, towards this perspective. Hopefully we'll be successful, but it's only going to be successful if the community gets behind it and not probably not just the science community, but the decision-making community. So that's a long-winded answer to a very good question. I'm sorry for skirting around that one, but only time will tell. All right. We're rooting for you too. Great.
Thank you for that, Bob. I have a question about your perspective from the outside. Do you think the limits to our collaboration are money, or trust, or something else? Like, what do you think is what's holding back this larger vision?
Lisa Marie, that's a question that let me answer that as an individual. I've been part of programs elsewhere that I think are far more advanced than what I see in the Delta. I'm not speaking as a Delta ISB member right now. And the programs to me that have been most successful were ones that we had in both, well, in particular in South Africa, is that we had teams of people, and it was everybody from the people on the ground, the game guards and so on, out on the ground, right up to the administrators within the South African National Park system, that when issues would arise, these teams were called together and they literally truly worked as a trusted team, whereby whether you were a game guard or a scientist or a technician or whatever, right up to the head of particular parks, you sat in a room until that issue was resolved. And you did that as an integrated team, and you stayed together. You didn't move from agency to agency or from town to town. These people were there for long periods of time. They knew each other as families and as individuals and so forth. And then they would solve the problem together. And I don't see us doing that here. I bring this up a lot. For years, I taught watershed management at the University of Washington, and I was deeply involved in, Pacific Northwest issues along these lines. And we always had these systemic problems, even in the Northwest, whether it was salmon or spotted owls or whatever. We were always thinking money was the answer. Money wasn't the answer. It was people, and how people interacted as integrated teams from the top to the bottom.
Okay, so let's thank the speaker, and we have a break until 11:35, right? Yes. You can keep talking.
Picture questions that people come up with as they're listening to these talks, and then we can use those to sort them out. So if you think of something, wow, does this talk address this or that other topic, please scan the QR code and type that in.
Okay, well, I'm gonna get started here. So thanks again for coming, everyone. I have to say I'm really impressed by the quality of speakers that we've had so far. It's really gotten me to think a little bit outside of my silo. And so I'm looking forward to the next round here. We're gonna switch gears a little bit and move towards empirical studies, in some case, sort of case studies, the stuff that is the fuel for the modeling that we've been thinking about over the past session here. And so we have 3 speakers in this session I'd like to introduce. We'll start off with Brian Bergamaschi from the California Water Science Center at the USGS, who is going to talk about phytoplankton and how we fix that problem, apparently. Brian has a PhD from the University of Washington. He's currently been working at the USGS on nutrients and primary production, especially in the freshwater portion of the estuary. And so he'll be focusing on that subject. He'll be followed by Kyle Phillips, who is a UC Davis candidate for the PhD, soon to be graduating, who has been working on primary and secondary production in managed wetlands in the sort of brackish to fresh portion of the San Francisco estuary. And then finally, we'll have Abby Ward, who is a research specialist at the Center for Watershed Sciences here at UC Davis, and who recently got accepted to a PhD program at UC Santa Cruz, right? Yeah, congratulations. So she's going to be taking us all the way out to the ocean, looking at thymine effects and the anchovy transition in the marine food web and how that affects many layers of the estuarine marine system. And so, Brian, I got you queued up here. I have no idea how this pointer works, but let's. So it's a gyroscopic pointer. So you press and you have to kind of. Okay. Oh, cool. Okay. So good luck with it. All right. Thanks. Okay. Oh, I need it. Thanks. So I'm not hyper caffeinated. I just talk really fast. Okay. So I thought John asked me to talk very broadly, not about one specific study, about nutrients in phytoplankton, what the background and history, and a little bit about where our recent research is pointing to. I want to acknowledge my co-authors, of course, and all the people that fund the work that we do. I'm going to present data kind of from 3 different parts of our program here. We've got this on the left, gyroscopic. On the left, we've got what we call high-resolution water quality mapping surveys. We built out this boat here that can operate at high speeds in very shallow waters, and we essentially set up a chemistry lab on the boat using instrumentation that can take measurements at about once per second, so we can make highly spatially resolved surveys across broad areas. And the reason you want to do that is you're trying to freeze everything in the tidal frame. We also are going to present some data from our continuous station network. We've got continuous monitors for a lot of water quality variables, including nitrate, which is important in the system. And then we've recently taken up Jim Klern's Bay water quality program, which has been recording across the transect through the Bay since the 1970s. So that's the kind of data you're going to see. We're talking about the kind of the contradiction, and we're gonna do both halves of the contradiction and phytoplankton nutrient story. Of course, we all know that light and nutrients are necessary to make phytoplankton, but then the story kind of diverges. What we really want to restore our aquatic ecosystems is we want the right kind of phytoplankton to make the right kind of zooplankton to feed the right kind of fish. You guys decide who's right. I don't know. I'm a chemist, by the way. Biology frustrates me. So we call these beneficial algal blooms. But, you know, it can go the other way. Even some of the same phytoplankton, this is a very strange point, can make nuisance blooms or toxic blooms, and we call those harmful algal blooms. And we're going to go there first. But a little kind of putting the San Francisco ecosystem in the context of estuaries across the world. There's some great compilations by Jim Klern. We are the largest estuary on the Pacific coast. In terms of the nitrogen loading, for example, we're pretty high with respect to the world's estuaries. In terms of phytoplankton productivity, we're kind of in the middle, and we generally have pretty low standing stocks of chlorophyll. And the reason we can have high nutrients and kind of low production in chlorophyll is thought because we've got this turbidity, restricting light, strong tidal mixing, and grazing by clams. So jumping first off to the HAB story, HABs up in the freshwater reaches are dominated, of course, by cyanoHABs. Cyanobacteria, principally, the focus has been on microcystis, and the toxin that it's formed is microcystin. And these have been observed in the Delta. They're not new. They've been observed since 1913. Work by Peggy Lehman and others have shown that they appear to be increasing in frequency, and we've got increasing frequency of detections of the toxins. This is thought to be related to the nutrients. We really haven't got a good handle on even the occurrence and extent of these organisms yet. They're designing monitoring programs right now. However,
down further in the bay, we saw a very, very large HAB event in August of 2022. We went for historic proportions. We broke a lot of records here. So over here is the long-term data from this long-term monitoring program. So the blue box plots are the chlorophyll concentrations by month for all the data between 1980 and 2021. And then the brown are box plots by month for 2022. And here's when that bloom occurred. You can see that it just was off the charts. You can see this thing from the satellite. It started about the first week of August, went screaming across the bay through August, up into San Pablo Bay. It was quite interesting that it didn't come up into Suisun Bay. We're looking and trying to understand why, but it screamed all across the bay and then just completely died out almost instantaneously. Very cool bloom. But I got to say, on a personal level, it was really weird to be out there sampling this thing. I mean, it was like walking through, a raging wildfire. We were doing profiles where we had no measurable oxygen from the top of the water to the bottom. It was just crazy. Never seen anything like it. So we were out there doing our mapping surveys here in our boat. Yeah, I should. Out here on our boat. So we were out there in July. We typically do these about once a month, but we were out there in the first week of August seeing the bloom start. And you can see that where the bloom starts. So we've on the top row, we've got the chlorophyll maps. On the bottom row, we've got the nitrate maps. Where you start to see the bloom come up, you start to see the nitrate completely wiped out. And on the 17th, you see the bloom extend in our mapping surveys. And until on the 25th, we couldn't measure any nitrate in the bay. It was just stunning. And then on the 31st, out there from the boat, it wasn't just the satellite that couldn't see the chlorophyll. We couldn't find the chlorophyll measuring in the water. It was wild. But I do want to point out to you, because I'm going to come back to it in a second, that in July, we had a situation where we had a little bloom starting right here. Oh, wow, that's cool. Oh, look at that. But it also depleted the nitrate stocks down here. Now, when we look at the data from a shoal buoy we have out lower in the bay, you can see that the bloom reached this location in the middle of August and climbed steadily. But this bloom, you can also see this is the nitrate. It crashed immediately. So just at the start of this bloom, it consumed all the available resources of nitrate. And then the bloom was sustained by recycling and continued discharge from the wastewater treatment plants. So, was that a black swan event? It's just a wild thing that we're never going to see that again. How many sturgeon died, Jim? 937. Rays, it was, anyway, wild. So is that a black swan event? I'm going to tell you what I think. So this is one of our water quality mapping surveys here where we were zigzagging across the bay. This is from the prior year. And I'm going to focus in on this one transect across the bay, which I boxed along here. So this is the time series of our boat. This is nitrate up here. It's running at 40 micromole. We've got a ton of nitrate out there, right? We hit the edge of this bloom within about 10 meters. The nitrate goes to 0, to undetectable levels, and chlorophyll concentrations go up to 40 or 50, sometimes 100. This is something, so we get out there about once a month, but this is something we see about a third of the time we go out there. We are leaving a burning candle with this in the middle of San Francisco Bay, just waiting for the right conditions to crop up to let it expand. So it's something I think we ought to pay attention to. Oh, by the way, so I'm going to do a little thing like Bob did with, that's my own opinion. It's not the opinion of the USGS, just to be clear. Not for citation or attribution. All right, so now we're going to worry about BABs, beneficial algal blooms, which are an issue up in Suisun Bay. Here's one of these iconic plots that we've got. We saw that this is chlorophyll and model primary productivity. We saw that it disappeared in mid 80s. So you need, of course, you need phytoplankton to have primary production. So the primary production crashed, and it's thought to have contributed to the decline in the fish species that we're trying to save. But there's good news. In recent years, we've seen that there's evidence that some of the blooms are trying to come back, and we were really interested in that. Now, can we figure out why? Can we do anything about it? We're kind of diving into this question more recently. But let me go back in time a little bit. Why did this blooms disappear in the first place? Many, if not most people, are quite convinced that it's related to the invasion of the Asian clam in the system. Patema corbula, which invaded, the invasion was documented in 1986 here at the yellow arrow. Come to that blue arrow in a second. When this is a paper by Lisa Lucas, where she showed that the clam grazing intensity is related to the disappearance of chlorophyll in the system. You can even see little hints here where the grazing pressure is low, that you might even get a little bit of rebound in the chlorophyll standing stocks. Now, this is from the paper by Fred Nichols and others, looking at where he said that he was looking at the invasion of this clam. This was a paper documenting the invasion of this clam, which happened in 1986, following a period of low river flows. And he posited in the paper that it expanded, this clam expanded because of these low river flows at the time. And he said that, you know, if we got high discharges, we might see some rebound in the benthic communities and some recovery. So, and we see that in later work by Jan Thompson and Francis Parchezo, where in wet years, we see the grazing pressure change in location, and we see it change. So this is the blue is the Patema corbula. And then after a particularly wet year, we see that the biomass of Patema corbula in blue was significantly attenuated. So turns out we did have some wet years. So we can test Fred and company's hypothesis. If you're not from around here, then you don't know that we had 2 of the wettest 5 years in Northern California in 2017 and 2019. This is the discharge in the Sacramento River colored by water year. So it starts on October 1 to September 30th. And we can look at that long-term record from the Bay Water Quality Program, and you can see that we don't see a rebound associated with those wetter years. But these are monthly surveys. You can miss a lot with the monthly survey. We don't have the continuous record for very long, but we were out there since 2018 at 2 sites in the Bay with some of our stations in Suisun Bay and Grizzly Bay, and down here what we call it the confluence. And you can see that we don't see much increase in the frequency or standing stock of chlorophyll in the system following these wet years. So there's another theory that some scientists subscribe to about why the blooms decline. It was related to the consolidation of wastewater treatment in the Sacramento area. This is the Regional Sand Wastewater Treatment Plant. And that was, that consolidation occurred in 1983, as I understand it. It's up here on the discharge point is up here on the Sacramento River, but it's the largest source of nitrogen to the Delta system. So the thinking here, if it sounds kind of confusing as to why adding a little bit more nutrient to the system would change the phytoplankton, I'll explain it to you here. So this is a an idea advanced by Francis Wilkerson, Dick Dugdale, and others. And what they noticed from a variety of mesocosm studies is you get the various highest growth rates and growth rates of the kinds of phytoplankton you want only after you consume the ammonium in the system, and then you can get these high production rates. And because of that effect, you tend to favor different phytoplankton at different nutrient concentrations and ratios, and that's what's shown here. So under the conditions that existed prior to 2021, the thinking was that we had high ammonium that led to, that favored the production of blue-green algae. Now, I am familiar that this is a controversial idea and that there's many algal physiologists who think this is nuts, but literally. But I got to say, I've looked at the experimental data they have, and it, you know, it's compelling as well. However, we'll look at our data here. So the phytoplankton biomass is low, where ammonium is high, and vice versa. So that's just the relationship that we observe. There's something like 200,000 data points shown. On the left, so what's plotted here is ammonium concentration on the x-axis for both, and this is the chlorophyll, and the chlorophyll that's attributed to diatom specifically on the y-axis. So in both cases, across several years, spanning, well, before and after the thing I'm going to talk about next, we see that where ammonium, where we see high standing stocks of chlorophyll or diatoms, we see low ammonium. So, but they fixed it. So, and this was great. I mean, this is a huge public works project. Regional SAN implemented the EcoWater project, and they changed their treatment system to essentially eliminate ammonium. This is the effluent concentrations in the effluent, just focusing on the blue, that's the ammonium. You can see that they brought their plant on line in 2 phases, and they completely eliminated ammonium in their discharge, or almost completely, and they reduced their nitrogenous waste by about 3/4 or so, better than they thought they would. Now, the bars here are what I'm going to show you next. These correspond to these high resolution water quality mapping surveys we do out there. These are the times. So we were out there before the year they were engaging in the first phase of the implementation, after the implementation. So I'm going to show you water quality mapping surveys from these years. It gets kind of complicated kind of fast, so we're going to focus only on spring. Plotted here, the ammonium concentrations, the nitrate concentration, the dissolved inorganic nitrogen concentrations. These data are all available on the web. You can get them whenever you want. Just looking at the ammonium concentrations in the system, you can see that particularly up here, there's whole reaches of the delta that have no measurable ammonium in them. That's changed. Out in the estuary here, the story is a little bit fuzzy. Just want to make the point that these surveys were conducted under similar hydrographic conditions, so we're not looking at dilution effects. So we're looking now at the different surveys, all the surveys by box plot, and we're looking at them by region, and the green bars when the full implementation happened. So looking up here in the Cache Slough complex, we can see a market effect of the reduction in ammonium. We get very low ammoniums up in that area, very low ammonium up in that area after the full implementation. In terms of nitrate, the story is a little bit fuzzier. In terms of chlorophyll, we see no systematic response. Looking here in the confluence region, we see that the ammonium is reduced. Nitrate story is complicated. Chlorophyll, we saw no systematic response in chlorophyll. Further out in Suisun Bay, we see that there is no particular significant systematic response in the ammonium concentrations in this data. Nitrates complicated, no particular change in the chlorophyll. All right. Well, you know, we go out 3 times a year. Really, is that enough to measure a system? So we're going to look at the high frequency data that we have. Here's the green bars where the wastewater upgrade was brought online. We see for these 2 sites no change like we did for the clam thing. We see no particular change in the frequency or the standing stock of chlorophyll or the frequency of blooms. So this kind of turned us into, you know, you've read about or seen the shows about storm chasers. Well, we're bloom chasers, okay? And it's way harder to be a bloom chaser because you can't forecast them, okay? So we got to see them, we got to run down to the boat, jump in and get out. But our leading hypotheses right now are that blooms are initiated by shallow water interactions that are driven by specific physical dynamics, and then separate physical dynamics allow these blooms to expand. So I'm going to give you a few examples of that. One of these is that we have observed that the spring bloom pretty much reliably occurs when X2, this is the 2 part per thousand, bottom salinity hits 65 kilometers from San Francisco Bay Bridge. So then you can see this is below normal wet year. Below several years, we see this bloom when it kicks off right when we see the 2 part per thousand hit this. And this is a marker for a physical phenomenon. When this salinity front hits it, it changes the physical dynamics of the main stem, but also the physical dynamics of the interaction with the shallows. We also see that interaction with the shallows and the Mars system also can kick off blooms. This is one of those maps where we can see a bloom up here in Grizzly Bay that gets to 50 micrograms per liter or so chlorophyll and significantly attenuates the nitrate standing stocks. We get out there 3 times a year, and we've seen these a ton of times. This is a phenomenon that happens a lot across all seasons. Intriguingly, it happened during the Salinity Gates experiment. Intriguingly, it also happens at the other end of Montezuma Slough. I forgot to point out that Montezuma Slough connects on both sides to the Suisun Bay area and confluence, and the tidal asymmetry through here, typically when the Salinity Gates are not operating, will push material out of Montezuma Slough into the main stem of the lower Sacramento River. Oh, we're definitely going to wrap up. You never leave time for questions. That's the important part. So we also know from the our data from the first Mallard site, Lisa showed some data for them, that it's a pretty productive system. We drive the shallow water exchange tidally, and we see this very large export. There's a recent paper by Ed Gross and John Durand and others that show the shallow water exchange and the water age leads to higher chlorophyll that can then be exported into bigger and bigger channels. We suspect that that's the process that's happening here. And I'm a tidal wetland fan, so I'm gonna make you sit through this slide because it's one of my favorites. This is Jan Harfman's paper. If you're not familiar with it, it's a pretty cool paper, but it speaks to the importance of wetlands in my mind. Took water from Montezuma Slough, which in its initial concentration was about 95.5% or 99.5% autotrophic organism, carbon, autotrophic carbon, very small amount of vascular plant carbon. But when you measured what was in the guts of the copepods, you saw that the vascular plant component was about 65%. Yeah, Matt likes that. Intriguingly, so that's really interesting. So they're selectively feeding on this plant detrital component. But more intriguingly, when you combine the plant detritus and feeding experiments with some herbaceous marsh detrital material, you extend their survival significantly. So we have to look beyond just seeding phytoplankton, but we do need to look at the detrital component of wetland systems and other shallow water systems. Yeah.
Okay, let's thank the speaker and we need to go to the next one.
Thanks, Brian. That was a perfect segue. Your ending was a perfect segue for our next talk, I believe, which is Kyle Phillips also talking about what feeds copepods. And it's not showing up. Thanks for saving it. Oh. Scroll down. We'll have to scroll through it. Yeah. Okay. Here's the pointer of death. Start between.
I'm just gonna go point.
Yeah. Take the mic.
Take this with me. You can use the pointer here also.
Oh, okay.
Yeah.
Okay.
All right.
Great.
Is there a slideshow mode? Oh, yeah. Oh. It's a PDF file. So is there a slideshow mode on it?
Go to view.
Single page? Thanks. Yeah, I don't Mac. Good.
Let's go down. Press it on the bottom. Slideshow.
Oh, whoops.
Did it?
Oh, I guess that's also works.
That
Great.
works. Perfect.
All right. I take this because I'm gonna just go point at things. All right. Yeah. So I've also got a lot of slides and I'm also really caffeinated. So we're just gonna go. Big thanks to all these people and all these groups. I'd like to start by challenging this idea. Full disclosure, I'm kind of a novel ecosystems person. But I like to challenge this idea that, these working landscapes or maybe working wetlands in this case are broken ecosystems. They can be quite functional. All of these examples, we have California rice fields, Chinese mulberry dike ponds, and Hawaiian fish ponds. They're all new and ancient examples of managed wetlands, essentially, and they're all very productive for fish. And I work in managed wetlands in Sassoon Marsh. That's my little area, and that's my box that I'm in. But I'll consider looking into other boxes too. This is me as a second-year grad student, starting my graduate experience. It's been fun. So that's one of them. One of the wetlands I study at it's called Means Landing. It's about a 700-acre wetland. So these things are pretty big. The way these things operate is they are seasonally flooded typically. So they'll often be flooded in the fall, and then they'll run water through them in the summer dry months. Or they'll run water through them through the spring, then they'll draw them down, and then you'll have all these plants growing in the wetland during that dry period. And it's exchanged through these tide gates. So it's all tidally powered. Kind of a neat system. And the reason for all this is it's for ducks. People love hunting ducks in Sassoon Marsh, and they impounded a lot of wetlands to grow and attract ducks and hunt them. But then there's also, some of these lands are operated by, most of them are private, but some are also operated by agencies. And then the other thing is we're finding they're really, really productive habitats as well, just kind of like those other examples I gave earlier. So they're really hot.
when these wetlands are flooded, after about 60 days, we start to see a big copepod bloom, especially Acanthocyclops. And then following that, around the 120, 4-month mark, we'll get a lot of Daphnia magna. So that's what we've been seeing. And I've been interested in what the mechanism is, and we'll get to that. I'm kind of glossing over my field methods on this, but that's because I want to get to the underlying cause. So right as we saw, this is potentially important because we saw there's a lot from earlier talks. There's a ton of food web problems. I can kind of gloss over this, right? The main problem is phytoplankton have been depleted, and that affects zooplankton. This is kind of a bottom-up effect we're seeing, and then it's causing these fish crashes. Right, the likely drivers we think are clams. We talk about water exports, droughts, habitat conversion from marsh to ag, especially terrestrial ag, as opposed to things like rice fields. Rice fields might actually still benefit aquatic ecosystems. And then that kind of slough channelization where water is just being forced out to sea as quickly as possible. So that water's not hanging out long enough to really produce things. So these are the likely problems, as others have highlighted. And as a result, this place is just a bad restaurant for fish. It's not really producing food for them. So then enters Sassoon Marsh, the box I study. It's still a relatively productive system compared to the rest of the delta. And it's in that, brackish mixing zone, as we know. It's big. And today it's mostly made up of managed wetlands, and a few tidal areas. And there's a couple more tidal areas in here now. This is an older plot or older map. So now there's a couple more restorations in there. And then I'll point you to this box plot over on the right. This was our sampling effort last year. So when we're looking at density of copepods, one of the major zooplankton groups, we often see higher densities of copepods in the managed wetlands, relatively few in the tidal wetlands. So that just goes to, again, to reemphasize they're relatively productive systems. So yeah, I'm curious what drives them. Oh, and here's a picture, by the way, of a big Daphnia magna swarm. All those dots are Daphnia. So that's just me on my phone taking a photo of the water. An example of how crazy it can get in these managed wetlands. So what drives it? Is it water residence time? Is it the fact that the water is just hanging out in these wetlands for a whole season as opposed to on the tidal cycle? Is it relief from grazers? Right, especially clams. I don't think clams can colonize the seasonal wetlands because when they dry out, the clams die. Doesn't work with their life cycle. And then the other is it flooded? Is it the vegetation? Is it the flooding itself? Is it that flood pulse? And I suspect it could be a combination of all three of these, but I wanted to test that last one. I wanted to look at the vegetation. So when you're looking in a managed wetland, picture on this lower part of the frame, over to the right, there's a cross section of the managed wetland and then kind of the slough interface. In the managed wetlands, you'll get this mix of emergent plants like your tules, Phragmites, cattails, and then you'll also get all these forbs that grow in the summer months. And then when they're flooded, those all this stuff basically produces detritus that can be consumed by the food web. So in the managed wetlands, we get this combination of forbs and emergent plants. In the sloughs, because they're tidal, they kind of just support the emergent plants. And I wanted to just test the litter from these two plants to see how it affected the food web. So I did a little mesocosm study. I took, let's see, how do I put this plainly? I take leaves from different plants. I put them in buckets of water from Sassoon Marsh, and then I watch those plants rot. So I watch the material rot and I see how the plankton like it. And that's my job. Yeah, so I incubated material from 3 different emergent plants, 3 different forb plants. And I did this in a greenhouse, temperature buffered and pools. I added live zooplankton after 10 days, and then I just monitored those mesocosms. And then I also did a repeat of this experiment, not just using single species, but also combinations of species, because I was interested in that too. So the phytoplankton, I'll direct your attention to the top right plot. In the control treatment, which was just no added vegetation, we got a little bloom of chlorophyll early on, and then it immediately crashed. So just in those first few days, little tiny bloom. And the emergent groups, the 3 emergent plant species, similar magnitude of bloom, but longer. So it lasted a bit longer, but the forbs it went gangbusters. So the chlorophyll really took off in all 3 forb species. And then this lower plot is just it's a quadratic linear model to model the effect of those functional groups, forbs versus emergence. The zooplankton production followed suit. So in the 3 emergent plants is pretty low. The blue, that was pretty low and comparable to the control, but they took off in the forb group. There is a trade-off though with this that's important to highlight is DO effects, right? Basically, I made some really nasty tea for the zooplankton by just putting leaves in buckets. And in that first week, we did see, big dissolved oxygen crashes, right? That's expected. The plant material's rotting. The microbes are going nuts. But then just after that first week, and once you get into that second week, we found it kind of fixed itself purely through biological processes, no wind mixing or anything like that. I didn't have any mixing going on. So purely through biological processes, it kind of fixed itself after a week. So, but this is a double-edged sword, right, that we're playing with. So flooding these things, these plants can create toxic conditions, at least temporarily. But you give it a little bit of time and then it shakes out. So then this is looking at the effect of, right, I mentioned I did combinations of species too. And I found that increasing the number of species also benefits the plankton production. But also the composition, the more forbs you add, the stronger the benefit. So if you see here on this plot, these first 6 are single species. And obviously, when you have a single species, it can either be all forbs or not forbs. So it's either going to be forbs or it's emergent. When you have 3 species, then I can experiment with different percentages. But we see, like, as I'm adding more species, it's getting just a little bit higher, those peaks of Daphnia production in this case. And so when I construct a generalized additive mixed model to model this, we see up at the top here that, increasing the composition of forbs increases production or density. And then actually richness does too. I was a little surprised by that. I expected richness to not really matter. I thought it'd be entirely based on the functional composition, but it does seem to matter. So there must be like some micronutrients or something else that's also affecting the productivity. And then, we have these fun 3-dimensional plots. The one that's interesting to me here is as you increase forb composition and you increase species richness of the detritus, you get higher zooplankton densities. So the implications of this, right? The vegetation matters potentially, but I am curious how this would actually scale up in the field. This is just a mesocosm experiment, so it would be worthwhile to see how these things actually affect the productivity in the field. And then the other thing is managed wetlands are essentially novel brackish floodplains. They operate similarly to the rice fields and the natural floodplains. And we could potentially use them as that to boost production or food production for fish. But there are some trade-offs, of course, to consider. Like, again, with the DO, you kind of got to be careful with that. So kind of thinking again, like how might we fit managed wetlands into future wetland conservation? Well, one idea that we had, is you can take, in these tidally restored systems, we could essentially connect them to these managed wetlands that are in place. So an example here, there's the Tule Red restoration, which we've been monitoring this last year. And right next to it is Grizzly King, which is a managed wetland that we've also been monitoring this last year. This is the difference in copepod densities we've been seeing between the two over the year. And that's the water year of 2023. So that'd be October 2022 through September 2023. And one thought is like, well, you could just put a gate here at the bottom of it, and just when there is high production in the managed wetland, send that water out and subsidize the neighboring system to boost that productivity for fish. And that's all I got. Thanks, y'all.
Unfortunately, no time for questions, so we will jump to Abby Ward. Remember that you can submit your questions to the platform.
Hi, I'm going to move this down just slightly. Sorry. Hi, my name is Abby Ward, and I am a researcher here at the Center for Watershed Sciences. And I'm going to be talking a little bit today about some food web shifts that have been happening in our marine ecosystem and how that has been influencing thiamine deficiency in our California salmonids. Now, today I'm going to be talking a little bit on a broader scale. So I'm not going to be focusing on some of our fine-tuned research that we've done. So if you do have more specific questions on our research, feel free to put it in our chat or just come and find me afterwards or ask me during our panel. So thiamine deficiency was first realized in California salmonids back in 2020 when juveniles and hatcheries started to display this sort of corkscrew swimming pattern. They were really lethargic and laying on the bottoms of raceways, and we saw really high mortality rates in a lot of our hatcheries. And so U.S. Fish and Wildlife and California Department of Fish and Wildlife started to think that there might be a disease outbreak among a lot of our hatchery systems. But as they started to see this symptomatic expression, they started to recognize it as a thiamine or vitamin B1 deficiency. And so they started to put B1 into the water that these juveniles were in, and they actually were able to recover. So this started an entire treatment effort in a lot of our hatchery ecosystems where females were receiving thiamine injections, and we were implementing these egg bath treatments as well. And we started an entire multi-agency collaboration, not only across the state of California, but across the United States and even beyond as well. And this included this egg thiamine monitoring program where we started to look at the egg thiamine levels of all of the different hatcheries across the state of California. And we did this not only for Chinook, which you see in this graphic here, but we've also done this for our steelhead and our coho as well. And not just in the Central Valley, but also for our coastal populations. And you can see over here on the right, let me see if I can figure out this pointer. Hold the center. It's okay. I can use this. Okay, I'll use this. Okay, thank you. So you can see over here on the right that we've categorized these into different levels of impact. And we've done this by raising eggs from our 4 different runs to 120 days post-fertilization. And we've measured the egg thiamine levels of those individuals and looked at the mortality rates. And then we were able to model the egg thiamine levels based on their mortality. And so we've been able to use this model to determine the level of impact. And you can see here that from 2020 to 2023, we've just had increasing levels of impact across all of our populations, but specifically some of our most vulnerable populations, like our winter and spring run that you can see here at the top. So all of our populations are just increasing in impact. And this is not only true for our Chinook in the Central Valley, but this is true for our steelhead and our coho and all of our coastal populations as well. And thiamine cycling is actually a very complicated process, and I won't get into all the chemistry that you see on this slide, but thiamine biosynthesis actually happens at the base of our food web, where primary producers, bacteria and phytoplankton, produce our thiamine, and they biosynthesize it through two different pathways, and they are able to synthesize this, and then it's moving up the food web. However, we have specific planktivorous fish that are producing an enzyme called thymidinase that is able to degrade our thiamine. And so whenever we have our predators that are consuming these specific prey items, they are then depleting their thiamine resources. And whenever they are consuming these specific prey items in high amounts, they are then making their thymidinase levels much higher and depleting their own thiamine resources. And this causes this reproductive influence where they are no longer able to allocate proper thiamine levels to their eggs. And this causes their juveniles to then be born thiamine deficient. And that causes lots of different effects. Like I mentioned before, this corkscrew swimming, high mortality rates, but it can actually cause really long-term effects as well, like impaired immune system, impaired development and growth. And all of these things are things that we have to consider whenever we're talking about thiamine deficiency. But as we started to consider this thiamine deficiency issue in our California salmonids, we started to recognize that our predators were not able to produce thiamine endogenously. They had to get it from their diet. And so we started to look at what could be disrupting their food web that could be introducing thiamine deficiency into our California salmon. And what we realized was that from 2017 to 2021, northern anchovy biomass and the northern end of their distribution changed drastically off the coast of California, where you can see here in the, sorry, I'm going to keep pointing on accident. You can see here in the green circles that they have just increased in abundance across, along the California coast. And you can see also from the 80s until now that the diet has significantly shifted in our California salmon, where in the 80s, our salmon diet used to be very diverse. Not only anchovies were included, but juvenile rockfish and krill and squid. But now it is very dominated by anchovies. And this is really significant because anchovies, out of all of the different prey items that our salmon consume in the ocean, are the highest, have the highest thymidinase activity. And not only that, but they have the lowest thiamine concentrations and they have the highest lipid content as well. And this is actually another driver of thiamine deficiency, because this can be an inducer of oxidative stress, which can be another driver of thiamine deficiency. But actually, food web disruptions and thiamine deficiency are a very common story. We see this with M74 syndrome in the Baltic. M74 is just another name for thiamine deficiency, so don't let that confuse you. But this has been going on since 1974 and in Atlantic salmon, and you can see how M74 has been correlated with very low thiamine levels throughout the history of the Baltic. And this has been due to sprat and herring, two thiaminase producers in this region. And in the Great Lakes as well, TDC has been occurring since the late 1970s and 80s, when alewife became even more abundant, an introduced species in this area. And you can see that high levels of alewife consumption correlates with very low thiamine availability. But moving away from fish, American alligators have also been seen to be thiamine deficient, as have eels and blue mussels as well. And Bulk's 2016 paper actually said that thiamine deficiency was a widespread issue across the Northern Hemisphere. And it's not something that's just limited to fish, but it's something that is been a known issue across many different species. And most recently in California, we've heard conversations around sea lion pup stillbirths, and brown pelicans that are showing up sick and dying. And both of these have very unknown causes at this point. And there has been research being poured into understanding if thiamine deficiency is actually a cause in either of these problems. And we don't know what the causes are right now, but we are now having to have more discussions on if thiamine deficiency is actually moving up the food web in new and unexpected ways. But how do we determine the diet of our salmon? How do we know if their diet has shifted and changed over time? We certainly can't follow them around in the ocean and try to see what they're eating on a day-to-day basis. But we are able to take certain tissues once they are back in the hatcheries and look at the stable isotopes in those tissues to try to determine their diet while they were in the ocean. We do have the ability, like I showed earlier, to look at gut content data, but gut content data can be really limited by our access to certain runs and populations and can bias us towards certain populations that we have access to at that time. So stable isotopes give us more of a broader opportunity to access different populations that we might not have the opportunity to get gut content data for. And so we can look at stable isotopes in different tissues such as muscle or eye lenses or scales. I'll be focusing on the stable isotopes of the eggs that I have also showed earlier that we have thiamine data for. So here I'm just showing a carbon and nitrogen biplot that shows the mean values for various prey and predator species within the California marine food web and focusing on where different main forage species of our salmon lie. And through measuring the stable isotopes of these prey items annually, we can understand interannual variability and spatial variability as well, and then be able to determine the different prey items that our salmon are consuming at different points in time. So here I'm showing another carbon and nitrogen biplot that is looking at the carbon and nitrogen in the eggs that we have also measured thiamine levels in 2020 and 2021. This is specifically for our Chinook rather than any of the other species that I mentioned that we also looked at thiamine levels in. And here we have divided our Central Valley into our circles here and then our coastal into our squares. And what you can see is that our circles, our Central Valley stocks, are tending to sit much higher trophically compared to our coastal stocks, which are sitting much lower trophically. And this is really indicative of specific prey items. So it shows that our coastal stocks are tending to eat much lower trophically, such as with krill and juvenile rockfish, whereas our Central Valley stocks are eating much higher trophically, such as with our anchovies. And then we were able to estimate our specific niche spaces of our different runs and hatcheries by looking at the ellipse spaces that these different runs were the ellipse area of the runs compared to the percent mortality that we used based on the model that I described earlier. And what you can see here is that our coastal Chinook ellipse areas were significantly larger than our Central Valley Chinook. And not only that, but the predicted mortality rates for our coastal stocks are much lower compared to the percent mortality for our Central Valley stocks. And this is really significant considering what these two different stocks would be potentially consuming. That if our coastal stocks were consuming a much more, a wider and more diverse diet compared to our Central Valley stocks, which would be considered to consume a much narrower diet, maybe based on anchovies rather than a broader diet on rockfish or krill or squid, could be driving thiamine deficiency in these populations. And this becomes even more significant whenever we look at the thiamine levels of these two different populations. Whenever we look at the thiamine levels of our Central Valley and our coastal stocks, we see that our Central Valley is significantly more thiamine deficient than our coastal stocks. And we can see this clear differentiation where our Central Valley stocks are eating much higher trophically with much lower thiamine levels, whereas our coastal stocks are eating much lower trophically with much higher thiamine levels overall. And this is really significant whenever we are trying to understand mitigation efforts and understand the diet consumption of our individual stocks and understanding how to mitigate differently in different populations. So we do have different mitigation efforts and different management strategies that we have attempted to implement in our hatchery systems. So we have two different runs, our winter and our spring run, that enter into our systems a little bit earlier before they have finished allocating all of their resources into their eggs. And this gives us an opportunity for early intervention. So we are able to interact with those individuals earlier, inject them with a thiamine injection, and then they are able to still allocate that thiamine into their eggs. And so you can see that we've had really high success with those treatments. Meanwhile, for our fall run, we've implemented an egg bath treatment where we as they are being spawned, we add thiamine into those egg baths. And we've had really high success rates with our fall run as well. And so this is actually implemented not just with our fall run, but this has been implemented in some of our steelhead hatcheries as well. And we've seen some success in our steelhead populations as well. We are continuing to understand more about treatment efforts and figuring out new ways to treat our different populations in different ways. Again, you've seen that our coastal and Central Valley populations obviously have different levels of thiamine that they are experiencing. And so understanding how to implement mitigation efforts in different ways is really important. And that's something that we are still trying to understand better. However, our monitoring and mitigation can only go so far. These treatments are obviously very important and they target populations that are severely impacted by thiamine deficiency, but our access is very limited by our availability to come into contact with these fish. So that means our hatchery populations. But what about our wild populations? We have very little ways to understand how thiamine deficiency is actually impacting our wild populations, other than monitoring when we do have wild individuals come into our hatchery systems. But monitoring symptomatic expression and mortality rates is not as easily done for our wild individuals. So we end up supplementing our hatchery populations as we are treating our hatcheries and not giving that same supplementation to our wild individuals. But this has had us start rethinking a lot of fish health. As I mentioned before, our individuals that are expressing this symptomatic expression, when they are juveniles, they have this kind of corkscrew swimming pattern. They are really lethargic, but not all of our individuals with low thiamine necessarily express these symptomatic expressions. And they can make it past the juvenile mortality state and still have low thiamine, but they may look completely healthy. And this is something that we've had to decouple, is this idea of somatic health and understanding, especially for our wild populations, how to understand what thiamine deficiency could look like in our wild populations. Not only that, but we've had to start really considering adaptive capacity of these species and understanding how treatment could be influencing adaptive capacity in ways that we aren't expecting. And that's a lot of our research now is starting to be poured into adaptive capacity of our different salmon populations to thiamine deficiency. But we've also been considering the microbial influence to thiamine in the wild. I mentioned before that we have these primary producers that are the ones that are biosynthesizing thiamine. And we have been considering that there is the potential for thiamine to be readily available in wild ecosystems. There could be an abundance of thiamine in our freshwater ecosystems that our wild populations have access to, that our hatchery populations do not. And so measuring the thiamine availability in our freshwater ecosystems is going to be very important as we continue this research. Not only that, but understanding if maybe there is an entire shift in the basis of the food web that is causing this thiamine deficiency issue to begin with. But just as a whole, understanding that diet has a huge impact on reproductive health, not only just having enough food, but exactly what you are eating can have really, really big impacts on the health of your offspring. And that's something that we have just all began to understand in a really new and different way. And I'm sure that you all, as you saw the first slide, were wondering why only my name was up there, because obviously I could not have done all this research on my own, and you are entirely correct. We have a very, very extensive team. I really loved Bob's talk on just bringing everybody together in a really communal way. I think that Rachel Johnson and Nate Mantua have done a really wonderful job in bringing this team together and partnering everybody in this way. So yeah, we have a very expansive team that's working on all of the different pieces of this together. So yeah, if anybody has any questions, I don't know if we have time for questions, but.
We have time for one question.
Can you share what's known about the anchovy population on the coast? What's driving their expansion or increase in abundance, and whether that's being studied by your team?
Yes, so that is being studied primarily by NOAA. So that is one of our partners in this research. But I did not put this in my talk, but there have been many warm blobs, as I'm sure we all know, that have occurred in the ocean over the last several years. And so ocean warming has been a big consideration in why the anchovy population has shifted so much. Not only that, but there previously was a specific concept of anchovy boom and bust cycles that people assumed were very consistent, that now is being thought about in a very new and different way. There was a paper that came out, I think, two or 3 years ago now, that I can send to you if you would like to read it, about how anchovy regimes have shifted quite a bit. But yeah, it has to do with partially the warming of the ocean, but yeah, a lot of things that have changed in the ocean over the last several years. But that there's a lot of research being poured into that, specifically at NOAA. But that might just be my perspective, because that is a lot of what I am connected to is at NOAA.
It's my understanding that they are adapting to warming.
Yeah.
So it's a surprise, right?
Yes, it is a surprise.
Yeah, cool.
Yeah.
Okay, so with that, we close the morning. We're going to have this working lunch with the speakers and the organizers. We have several options, walking distance, so it's Segundo dining room and the Memorial Union. Yes, and please be back at 1:40 for the session 3, management strategies. And remember to submit questions if you haven't.